So everything's under visualizing genomics data in R. You notice it's all compiled yesterday or this morning. It is failing, but it's only failing for one of them, I think, and that's because it's a GitHub problem and not a system problem. So the code should be working for you. Yeah. And so hopefully everyone's installed their requirements. You should all have the majority of these packages now. There are a few new ones which we haven't covered before, like MBclust, Profile Plier, VSN, and Gfiz. Okay, so we're going to start off with the slides. And here we go. So obviously, visualizing your data, it's a massively important step. It allows you to look for both at QC levels, to look for things that are unexpected and potential artifacts, but then also to find the trends that you actually care about in your data. Not that you don't care about the bad stuff, the QC stuff, but it helps you find the stuff that you're actually excited about, new patterns, things you weren't expecting, but actually are biological. And so what we've done so far is we've discussed how to use IGV to do this, but we're going to now focus on how to do it in a programmatic way with R. So this is really, it's a complement to our original course that we did on IGV. So I think most of you have either there or have done it in the past. But you can always go back and double check how to do some of these things in IGV. But basically, in principle, in the past, we've used IGV. We load in things like bigwigs, and we can see patterns over genes, and that's nice. And then you can export it as an SVG, or basically export it as an image, and then you can load it into Illustrator or something like that, and you can make it look pretty and get it ready for your publication. But now that you all have these R skills, you know how plotting works in R, and you know how things like G ranges work in R, you can just do it all in R. And then you can have much tighter control, and you can do it much quicker for many, many gene regions. So then you can pick your favorite once you've actually done some things to make it like your the final aesthetic you want it to be in your paper. And so we'll be dealing with a bunch of different data types. We pretty much run the gamut on all the different data types we've talked about, but you can always go revisit what we've talked about. If you follow the link, go to file formats or IGV for how to use them with the IGV. And we have links to pretty much all the material which we'll touch upon, which is almost the entire course. But you can follow the links here if you want to check back in to one of these individual things. And so this is just a nice hub or shortcut, so you can hop back there if you're confused at any point. And as always, you can download the material. We find it on the GitHub, or you can follow the download. I think these download links might have actually stopped work. Oh no, this one works. Okay, I'll cancel that download. So yeah, you can download it, and then everything that you need you'll be able to find within the R course folder. And all the data that we'll be using is under the data directory. And you just need to set your path to be in the download in the name of the folder that you downloaded. You'll have to extract it first. And then you'll want to reposition your working directory to be within our course. Okay. And so this is the same as what we've been doing with every session. Okay, so I think this is clear at this point because I've explained it like 3 times, but why are we here, right? We've talked about IGV. IGV is so user-friendly, and it is initially very much easier to use IGV to extract your tracks. You have regions you care about, you just save them, super easy. But when you start caring about many regions, or say you want to present to someone several options for what to include in your final paper, it can actually be much easier to just do it programmatically rather than pointing and clicking and then going through each one in a very time consuming manner. And so you can invest that time in learning these Gfiz skills, and then the next time you have to do it a year from now, you already have code and script that can just do it, and you'll just have to change the inputs. And so, yeah. Yeah, this is pretty much all I've just said. So it provides it is an essential tool, and it is always our first pass when we look at the data. What we're going to talk about today won't provide you the ability to just scroll around the genome. It's much more targeted. So once you've found regions that you care about, or say differential genes, differential peaks, whatever top hits, that's when you can use the skills we learned today, or we'll learn today, to be able to extract regions you care about. Which is exactly here. So this is a critical first step, using a browser, and then we can move on if we have a large number of sites we care about. And this is something I haven't really hammered home, is it doesn't offer this flexibility. So you can export it from IGV, but then you have to spend time in Illustrator making it look like what you want it to look, to make it fit with the rest of your paper. And we can just do this in R, right? And then once you've set those parameters, it'll apply it to all of them, as opposed to having to manually customize everything. And so we do all this with the Gfiz package. And so first off, we need to install Gfiz. It's the same as we've done all along. Gfiz is a bioconductor package. So you can use Bioc Manager and the install function. And so the way that Gfiz works is you build up a series of tracks, and then you can visualize them all at the same time. And so the simplest track is just an axis. Okay, so this will be whatever you're labeling your data with. And it's a nice linear axis, and to do that you use the genome axis track function. Okay, and so with Gfiz, we create a track this way. We haven't given it really any information. We just know we've created this axis. And so when we look in genome axis, this object we created, all that's the only attribute it has at the moment is a name, which we provided here in the function. But it doesn't really have anything else. And so it knows from the fact that we've created an axis, it knows that we want to make an axis. But it doesn't render anything. It doesn't make a plot until you use the function plot tracks. Okay. And so plot tracks is a function we're going to keep coming back to. What we're going to be doing is keep making different kinds of tracks, and then we'll plot them with plot tracks. And we can plot them together or separately with plot tracks. It's all about what we put into this function. And so when we do plot tracks, we say we have an axis, we want to use an axis, and then we give it a range, okay? And so this is important for plot tracks because we need to provide information about where we care about. And so in this case, we care about from 100 to 10.1 kb. Okay. And because the only information at the moment we have is that we want an axis, it will make us an axis covering that region. Okay. So the only information we have stored in this genome axis track, if we remember, is a name. But because it knows it's an axis, it can infer, because it's assuming this is for biological data, right? It's Gfiz, genome visualization package. It's going to know what kind of axis we want. It's going to be KB based, and we can use the plot tracks to really set the limits. And so that's just kind of the start. And really what we're going to be doing is showing all the different ways you can customize these things as we go. And so first off, we're going to outline how you customize an axis. And so I know we don't have any actual data here yet, but we'll build up to that. So I think you can see in those couple of steps, it's really straightforward to get to the point where you're actually making some tracks, and they actually look pretty reasonable, I think. And it's really easy to just then start to play around and customize it to fit what you're looking for. And so one thing you might want to do is label the directionality. So you can add either the 5 to 3' direction or the 3' to 5' direction by using these logicals with these parameters, add 5.3 or add 3.5. And you can see it's just added little labels on the end. You can also configure the resolution. However, it's kind of, as it mentions, it's a little bit blunt. You don't have as much control over these little ticks as maybe you'd want. But you can just use little ticks equals true, and it'll add some little ticks in between. Yeah, I think it's quite clear that this is not a great way to look at things at the moment. But depending on your ranges, it could be super useful. Now, this is a nice addition. Is if you notice, it alternates the labels to make sure there's space. But depending on your axis, you might have space on everything to be on the bottom. So it looks a lot more aesthetically pleasing if everything's in register with each other on one side. And you do that with the label pos parameter, and you can set it to below. You can also set it to above if you want to do above. Okay. And so the axis we've shown before is more of like a positional index, right? So that will show you directly where a coordinate is for a base pair or something like that. So you can actually see this gene is so far along this chromosome or something like that. What we might prefer to do is actually show a scale bar. And so the genomic position might be less important, but actually just having a relative scale would be more important for whatever you're looking at. And so you can just add in a new parameter for scale equals 1. And what this will do is it will actually just turn it into a scale bar, and it will say this region here, this is 10 kb. Right, because we're saying from 100 to 10.1 kb. So it does all the math for you, and it will show you that region. And then you can also show how control how much you want to show. So this is a really starts to get really useful and nuanced, and you maybe don't fully can see the power of it yet until you start playing with your own data sets. But then being able to add a scale bar, we'll be able to later on add some, say, genome annotations, some some gene models. That's the word I'm looking for. You'll be able to have some gene models. You'll be able to have some signal over those gene models, and then you'll be able to have a scale bar at the bottom, so that people know the size. And so to do this, you can just control the scale parameter. And so in this case, we were saying we only want 0.3 of the total scale. And so as we have a 10 KB scale, 0.3 of that is just 3 KB shown. And so you can see that the rest is plotted. And this is important for when we add extra tracks that will build up on top of this to create your final figure. And so you can also provide numbers greater than 1 to the scale. Okay. And so if you don't want to have to do the math to figure out that 3 KB is 0.3, if you provide a number greater than 1, what it will do is it will assume you're directly saying how much of the KB you want to show. And so in this case, we say, oh, we want to actually scale it to 2,500. So it will show you 2.5 KB. And so the scale parameter then just switches how it's working from a relative proportion to you directly encoding exactly how big you want the scale bar to be. Okay. So that was the scale bar, and that's how we make an axis. Okay. It's maybe the least exciting part of these plots, but it's actually one of the most important, right? So that everyone has a reference for what they're looking at. And so next we're moving on to regions of interest. Okay. So when we worked with IGV, we saw how to highlight regions of interest in the bar here by using things like, you can actually see the regions of interest button underneath. And these can be any location you care about. They could be peaks, they could be genes, and they could be exons. And they're really important for adding context to whichever genomic data you have. Okay, so again, we're building up from the ground up this figure. And so we have a scale now. And the next bit you probably most of the time will want is some kind of gene model on top of that. And so to be able to do this, we need to actually define what is a region of interest. Okay, and to do that, we define ranges in much the same way we've done for everything else. So you can use I ranges, or you could use G ranges. Both of them work. And if you follow the link here, you remember we created I ranges and then made them into G ranges. And so depending on what you're doing with it, both of them work pretty well. But to start with, we're just going to do a basic I range. And so the key part about an I range is you just have a start and an end. And it infers from that the width. So the big difference between an I range and a G range, if you remember, is a G range is genomic focus, so you will also have some sequence associated with it. And by a sequence, I mean a chromosome. And so the third column in a G range would be what chromosome does this region belong to. Okay, so we can load in the iRanges package, and then we can create our ranges of interest. So we're going to create 2 ranges. One will start at 140, one will start at 5140. Okay. And likewise, we have ends here. And we're also going to add some names to them. Okay, and so then when we just look at the iRanges, it will look like this. So we have the name associated, and we have the start end, and it's inferred the width. And so we have the same width of both of these. So we can now include our regions of interest onto our scale. And so what it's doing here is it's just going to highlight the scale in these regions that you care about. And so a lot of these different tracks will be able to interact with these regions of interest objects to subset them or modify them in some way. So in this case, we're going to recreate our genome axis track. But this time, instead of just providing the name argument, we're actually going to add a range. And so that goes into the range parameter here. Okay, and we're adding our regions of interest. And then we can plot tracks again. And now you can see that not too much has changed. The axis looks fairly similar, but we do have these highlighted regions, which show where your regions of interest are. And we can play around with this. So we can include labels. So we had names for this. And so we can include these labels within the axis plot by using the show ID parameter and setting that to true. Okay, so we're going show ID equals true. And the names are here. They're just very small. Okay, and so we'll talk about a little bit later about using controlling the size of things like this. But essentially, it will be the same as when we did basic plotting, where you use an argument called CEX to make things bigger. Yeah. And so with all this, you have some, like, hopefully you can see how you can control the axis. You can label parts of the axis. You can set it to a scale if you prefer. And you can really set a nice backdrop for the data that you're going to add. And so what we've been waiting for is to actually add some data. Okay. And so once you have the regions that you care about, you have that set up, you can then use this kind of general container that Gfiz has called data track. And so what data track is basically a special object that's specific to Gfiz, which you store in it just score information. And this score information can then be plotted in a way to using the coordinates. And so, yeah, you can see here it has some description of what data track works with. Most data types work. It tries to figure most things out. It's like most things in art, right? It will infer what it can and try its best. But what it works best is if you have ranges that have signal associated with it. As a matrix, if you have multiple regions, or a simple vector, if you have a single sample that you care about. So in this case, we can use our iRanges still, and we can use the mcols function, just as like we did for GRanges. And then we can update this to be a genomic range and add some scores basically to our range. And so how we're doing this is we're doing a data frame. we want sample 1 to have these scores and sample 2 to have these scores. And we're going to be adding it using the mcols function. Okay, so if you remember, the mcols is an accessor function that allows you to access the metadata of these range objects. And so we don't have any metadata yet, but what we're going to be doing is assigning this data frame into the metadata region. We're then going to use the granges function to turn our iRange into a grange. And so if you remember, the main difference is it has a chromosome associated with it. And so if we need that chromosome information, so in this case, we're going to provide it. And so that goes into the seq names argument. And we're going to say this is chromosome 5. And then we can put our iRanges from up here, which has the metadata. We just provide it to the ranges argument. Okay, and so now that iRanges has been turned into a grange, you have that additional information, and it will look like this. Okay, so it's very similar to what we had a couple slides ago, right? With start, end, and width, and these names. And so we have the start and the end contained within the ranges. We have the seq names. We have a column for strand, but we've provided no strand information yet. And then we have this metadata added on the end. And so now we can just use data track and plot the data track. So here we do data track on our grange. And then we do plot tracks on this object. And what you can see is the scores. So we had 30, 20, 20, 200. And so you can see the scores are being shown here. And we have the axis along here for the amplitude. Okay, so this is a nice, simple portrayal of some data in a data track. And so you've seen that they use, we can easily create this data track with a grange. And so that means anything we've done in granges before, we can take use of that before we create some kind of visualization. So we can modify and manipulate these granges in much the same way, and then generate new plots with that. And so in this case, we're going to use our track layer, and we're going to use the import tools, and we're going to use this to get coverage from a bigwig and have that as a granges object. So there's a small bigwig in the data directory. So hopefully you guys can do this as well. And you can use the import.bw function. And you specify what kind of object you want to create. So we're going to create a granges. I think if you leave this blank, it will create an RLE. So you want to make sure you specify this. And we're going to import it as all chromosome coverage. And so when you look at this, we can look at the class, and we've just created a granges here. And so what does this look like? So if you remember, with a granges and a bigwig, it will show a range for every score that's basically consistent. If that makes sense. So if you have, in this case, a score of 0, stretching the whole chromosome, it will show the whole chromosome. Okay, but if you have a score of 1 that is 1 base pair, that range will just be 1 base pair. And so we're actually focusing on chromosome 5 here, so that's not actually shown right here. But if you guys are looking at this object, you'll be able to subset all chromosome coverage to look at chromosome 5. Actually, let's just do that real quick. Okay, so I just imported it. It looks like that. And so we can do, no. So we can subset it to just chromosome 5 by doing so I'm going to call out the seq names functions, and I'm going to say it equals chromosome 5. Okay, and index based on that. Okay, and so you can see we have all the chromosome 5 regions, and for a good chunk at the beginning, there is no score. But then for a couple bases, yeah, like 30 bases, there's a score of 1, then it goes back down to 0, then it comes back up to 1, then it goes to 2, etc., etc. Okay, so it's just like an RLE list or an RLE, but it's just in a big a GRanges format. So yeah, we were just looking at that. And so now you have it as a GRanges object, we can actually just create the data track from it. Okay, so and we're going to do exactly what we did before, but this time we're actually going to specify the chromosome parameter. And I actually misspelled chromosome here, but it's still evaluated, but that's because we have some secret magic that we do when we compile. But obviously this should be spelled chromosome, not chromosome. But the key point here is that we can actually specify the chromosome that we care about. So this means instead of focusing on the entire genome, we'll just focus on the part that we care about. And then we can do plot tracks again. And we're going to give it the data track. And so this means it has access to the data in the whole of chromosome 5. But again, we're going to specify a region. Okay. And so these could be your coordinates that you care about, some gene, some genomic region. Maybe you want to plot a whole TAD. Or maybe you just want to plot just the promoter region. Okay, and so you can control it directly with these coordinates. And then what you get is this nice little dot plot. Okay, and so this is similar to what you get from IGV, right? But now it's reasonably aesthetically pleasing, and it's in R, so that means it's customizable in a way using the plotting tools that we've talked about in the past. And so it accepts a lot of different arguments for plotting tracks like this. And so you'll hopefully, from when we covered plotting in R, you'll recognize some of these terms. So like we can do type equals L, and it will turn into a line plot. We can do type equals smooth, and it will actually enact some smoothing. Okay, so it doesn't look quite so jumpy. We can do a histogram if we want. Okay, and so this will essentially be bars with lots of gaps as well. Or if we want to get a filled smooth, we can use something called a mountain. Okay, and we have this nice pink and blue. And lastly, you can even use heat map. Okay, and so this shows you the strength of the signal, but it's the dimensionality is inferred by the color instead of by the height on the axis. So depending on how much data you want to show, this can actually be a nice way of showing it if you need to show lots and lots of data next to each other. And yeah, we do point out that Gfiz will automatically produce the appropriate scale. So it doesn't just create some kind of buck-wild scale. It looks at the data and infers what's most appropriate. But that's pretty much the same with all of these plotting. It figures out the correct axis as well. And so, as with a lot of these things we mentioned before, you can customize them in much the same way. So you can use the CEX parameters and the col parameters. And it'll change, say here, we've changed the dots to be much larger and actually made the color red. Okay, so this is just kind of a taste of how you can start to customize these things. And put your own flavor into the plots that you're making. And this is one of the key strengths of doing it this way, right? Because if you wanted to do this with IGV method, you'd be exporting them all, loading them into Illustrator, and then changing this, but doing this for every single track you want to show. But with this, you can just repeat the code. And so as we start to build this up, you can add the axis in much the same way. So we have this axis here that we created earlier. And we have, we have the genome axis here that we created earlier. And then this is our data that we just made. Okay, so we have our data and we have our axis. And so we can still use plot tracks to plot both of them at the same time. We just supply all the tracks we want to plot at the same time in a vector. Okay, so we have our data and we've got the combine function sticking them together. And so now at the bottom here, we have the axis we created and we have our data at the top. And so depending on your order in the vector, that's what controls the order of the tracks, okay? And so quite often you'll want your axis at the bottom, but sometimes you might want to add the top. So you can just switch this around. I think this becomes most important when you have multiple data tracks. So maybe you have some, like a wild type and then a mutant and then a gene model and then the axis. Okay, and you want to display it in exactly that order. And so you can control it just right here by changing which order we supply the track in to plot tracks. So a lot of this is pretty smart with. It tries to provide sensible track heights. And so you can see things like the axis by default has quite a small track height because most of the time we don't need a gigantic axis, while the actual data is much larger because that's the point we normally care about. And so these relative sizes are generally pretty good out of the box, but if you want to actually add some fine-tune control of this, you can use the sizes parameter in plot tracks. And so when you have multiple tracks, you'll need to provide relative sizes for these multiple tracks. And so in this case, we want the axis to be 50% of the height of the data track, okay? So before it was a lot smaller. So you can see this is maybe 10%. So we're making it bigger. And so now it's going to be 1/3, okay? Because it's 0.5 to 1. And so the key thing about this is the order of the size represents the order up here. So you just have to make sure everything's relative. Okay. Now, I think this is my favorite part of Gfiz. It's when you start to add annotations, such as gene models and transcripts. I think this is the really a nice thing when you can have all of this together. We have a question. When you specify a color, it appears the axis takes the same color. Could that be changed? I believe so. I think each of the elements are customizable, but I have not done that. Let's see. Let's go on a Google together. Gfiz color. Okay. So the Gfiz user guide from Bioconductor. It looks like they did a pretty nice job. So you can see people adding chromosomes and stuff. That's pretty cool. So let's go to color. So you can see there's lots of different arguments for color, such as the border title, ID, things like that, fonts. But I do have an idea that might work. So I'm thinking that you could probably provide a vector of color as well. But I just haven't seen that in this help. So much like we did for size, let's give that a go. And if it doesn't work, then I'll have a little look later on, as this hasn't jumped out as an obvious answer. So I'm going to switch over to my RStudio. Okay, and so I just have my regions here. So let's plot some of this data that we were just looking at. I need to just define my data track. Oh, I actually need to load in Gfiz first. That'll be good. That means I also need an axis. So let's go back real quick and grab that from the slides. Okay. So we have our axis and we have, oh, you tried that earlier. Okay. So maybe I won't bother trying this right now then. And my other idea is that you can specify the color when you actually define the object. Let's see, can you provide a color parameter to this? So let's see if we can define, give it a call. Okay, so I just gave it the color red. Let's try and plot these together now. There you go. That's your answer. Okay, so I provided the color red in the original definition of the axis. And that's how you control the separate colors. Okay, so you have all these different elements. I think it's similar to ggplot, right? So if you remember with ggplot, you can add color arguments to the main ggplot aesthetic, or at each independent level, you can control the aesthetics for just that level. So if you want to do scale by alpha, you can control some of the scaling elements directly in that function as well for the aesthetic mappings. Yeah, and for the genomes. But that was a good question, and I learned something as well, which are my favorite questions. Okay, so yeah, the next kind of track we'll learn about is annotation tracks, and I think these are some of the most useful, because we already know how to, like, we could make plots that look like this already with ggplot, to be honest. You create a data frame with position, right? And we can have an axis that it doesn't look as pretty as this, but it will have an x-axis. But building up genes, annotated genes with stuff like isoforms and stuff like that is really hard to do if you're going to try and do it manually through graphing. And it's also, when you go from IGV, can also be quite challenging to get all the different isoforms that you care about in your SVG object at the end. And so this annotation track function allows you to do it in a relatively straightforward way, using, again, all the skills that you know already from Bioconductor. And so you can start off with just some regions that you want to annotate. This is the very base level of this, using G ranges. Okay, so we have some regions we care about. We create this G ranges for chromosome 5, and we say, yeah, we care about these regions from 10 to 300, 500, 800. We have this G range here. And so now we can provide some information. This is just like a blank G range, right? We can then plot these as they are, and it will I'll just show them together, okay, as they will show them as squares. But what we can then do as an additional step is add a grouping parameter to these. And so we're saying the first two are part of annotation 1, the second one is part of annotation 2, and the third one is part of annotation 3. And so what this actually does is it links the first two ranges together and links the fourth and fifth ranges together. And what you see, I hope, is very reminiscent of something like a gene model. You have these boxes, and then they're linked together by a line. And so this is very similar to exons and introns, the way that they're portrayed. And so you can see how easy it is to go from these very simple G ranges to something that is essentially a gene model. You can also add group annotation. Okay, so if, for example, you don't use AN1 and 1, but say gene symbol 1, gene symbol 2, gene symbol 3, whichever genes you're focusing on, you can then say you want to turn on group annotation, and it'll annotate all of these. Okay, so they all have their names associated with them. When we did this, we didn't create any strand information. So if you use the strand function on this G ranges, you can see it is all star. It has a length of 5, and the value is all star. So that means they're all unstranded. And so whenever you give it unstranded information, it makes these boxes like this. However, if you give it stranded information, you can add, it adds directionality. So in this case, we're adding, so we call the strand accessor again. We add strand information. So we're adding strand information to the first two and the 4 and 5. We do the same grouping information. And now you can see that the ones that have strand information have some arrows. And so we're using exactly the same tool set that we've kind of been going over. We build this annotation track, and then we plot it. We're using plot tracks. Okay. And so that's the kind of, at the base level, is the most rudimentary way to build up an annotation of a region you care about. Okay, and it shows you how to do this with just your basic G ranges. So this doesn't have to be genes. This could be peaks, right? It could be TAD boundaries. It could be all kinds of things that you want to label using a more traditional annotation format. And so if you're going to consider more traditional annotation format like a GFF, when we were using that in IGV, you saw there was an ability to control the display. And so there were several options, but they include things like collapsed, expanded, or squished. And so that really relates to how, say, if you have several transcripts for a gene, how they are then interpreted. Because sometimes you have 100 transcripts for a single gene, and you just, it's not feasible to show all of them at once, right? And so you have to collapse them down in some sensible way. And so if we go back to our example, we can use the stacking called squish. And this is actually the default. This seems to have changed. It shouldn't have this. I think there must be an edit in the code. Nope. But yeah, squished is basically the default. So, because generally what squish does is it optimizes based on the space. So it doesn't take up too much space, but it doesn't take up too little space. It's not going to collapse things unless it unless you don't want to. Oh, we have a comment from Yozan. Oh, yes, you're right. Here, the annotation has been dropped because it should be group annotation, not grouping annotation. Sorry. But that shows you, so in this case, the function still works. This often happens if you misspell parameters. It will still execute and it will run fine. It just won't do the thing in that argument because it doesn't know what that argument is anymore because it's misspelled. Okay, so I'll also make a note of that one as well. Thanks for catching all the typos. Okay. Yeah, so squished is the default, but you can control which ones you use. And so in this case, we can use the dense option. And you can see here the extra, I'm just going to call it an exon gene, whatever we've annotated, which was up here has now been squished down to here. But you can actually see it delineated as well. So you have the gray line, which helps you illustrate where it was. Okay, so this annotation track object can hold extra information. Okay, so once you've controlled how it looks, you can add extra information beyond just its name. And so we can do that using the feature function. And so at the moment, our object has no assigned features. Okay, and so we can do feature on our annotation track, and it will just say it's all unknown. And so this could be anything. Like we give the example here that you can label it as a messenger RNA, microRNA, rRNA, snow RNA, whatever you want to label it. But you can also just label it based on some other characteristics that you have. Is it a good transcript or a bad transcript? It's a very arbitrary line to draw, but maybe it's because of its expression level, or because this is a gene that changes in response to your drug, so it's one you care about. And so these are really nice things to add, because say you want to show something's happening specifically to a gene or a couple of genes, you could show a region and then have it highlighted which ones are good or bad, or changing, not changing, sensitive, not sensitive. Okay, and so we've labeled these features here as good and bad. And then when you comes to plotting them, you can then actually control aesthetics associated with them. So in this case, we're going to change the color to blue and red. And so it basically creates arguments within plot tracks that are the different features that you have. Okay, so you now have this option for an argument for good, which you can then control to be blue. Okay, and you can also add this extra. You also need to turn on feature annotation as well. Okay, and so that labels it as good as well. So we have lots of labels now. We have labels for the gene or the exons, and we have the labels for whether it's good or bad. Oh, sorry, let me turn this off. Okay, so I think we've shown you a bunch of information about how making these complex annotation tracks that are custom, coming from G ranges. Okay, and so this is again a nice, useful tool to annotate what you have going on. But there's a special class of annotation track for gene models, and that's called a gene region track. And so that takes a lot of the things that we just did with annotation track and like takes it to the next level, because it is completely specialized for gene models. And really it's because it's a restricted data type, it can then add extra parameters that are specially for this kind of data. So we have there's a small gene model set stored in Gfiz, so we can just start by looking at that. And so to get it, you can use the data function, and you say gene models, and what this does is this data is stored in Gfiz, and it just calls it into your environment so that you can use it. And then if we have a look at it, we can look at the first row, and it's a long intergenic non-coding RNA. And you can see it has all this annotation information. So we're specifically looking at one of the exons. We have transcript symbol gene information, and we obviously have its position because it's a range. And then if we actually look at the whole, a little bit more of this, you can see, yeah, it basically contains all this information within it. So it's super useful. And it's broken down by exon. So you can see this is actually all belongs to the same gene, It's not all the same transcript, but it's all the same gene. And so then we create this gene region track as we would any other track using its special function. But we can pass additional information. So we can actually say what genome we got it from. We can say what chromosome we care about. And we can provide a name. And when we do all this and do plot tracks, you can actually now see there's a really nice portrayal of the genome annotation. Or the gene annotation, not genome annotation, gene annotation. You can see all these models, right? And you can see this complex transcript makeup of this link RNA. And so you can see it's actually, it's rendered in a slightly different way to Annotation Track. You can see we have the intronic regions and they have arrows in them. And the untranslated regions are the narrower boxes. So that's the UTRs. Okay. And so this is really the standard convention for displaying gene models. And it has all of that within it. And you can see how easy it was, right? We just need our gene models and to use the gene region track to import it, and then we do plot tracks. As with what we did before, you can add annotation information. There's two parameters you want to focus on: transcript annotation and exon annotation. Okay, and so you can annotate each transcript with, say, if we use gene, it will give it the gene ID or whatever's in the gene column. So if we go back, so this is the ensemble gene ID. But you can use whichever column you want. So if you want symbol, transcript, or exon, whichever. So here we did the transcript annotation. And again, it's simple. And this is arbitrary. If you have extra annotation information, you can add it, whatever you want it to be. And we can also label individual exons if we want. So in this case, we're just focusing on one of the models by really focusing on a specific region. So let's see which one. I guess it must be one of these guys. Oh no, it's this one. We're focusing it in on this. Okay. We're doing that with the from and to. And you can add exon information as exon annotation. And so you can see it's small. But you can control all these things. But you have quick, easy exon annotation. And so as with Annotation Track, you can control the stacking. So in this case, we're saying dense. And so this is like the same as this. Okay. So it's got the same information in it, but the stacking's been dense, so it collapses it down in much the same way. It's a lot harder to interpret, I think, this way, but sometimes this is what your data calls for. So you just have to see where you're going to fit it. If you're going to have like a science paper where there's zero space in your figures, then you're going to have to go for something like this if you want to display a gene model, right? But it all depends on what you're doing. Maybe a more useful way to show it is to use the collapse transcript parameter. So this, then at this point, it doesn't really show you anything to do with the transcript information. It just shows you where the genes are. So SCAP2 was the biggest gene, right? And most of it was empty. Most of it was not empty. Most of it was intronic. But we can just put it as this one big block. And then we've done our transcript annotation as symbol, so we can see these symbols. And then you can also play around, especially when you do something like this. Actually, adding back the arrow format can be very useful. So you can add shape equals arrow. So you can get the directionality because you can't see the introns anymore. And so you can't see those little arrows on the intronic lines. And so then there are some other options. Maybe you want to collapse the transcripts, but you do want to display a transcript. You still want to have some intronic information. So you can still use collapse transcript, but instead of it just collapsing everything down to everything being displayed, you can select which one you want to display. So in this case, we're choosing the longest. The alternative is to collapse the transcripts down, and you can choose meta. So with meta, it shows you all the potential exons. This one's a nice one, I think, as it's this composite lossless illustration of gene models. You don't get an idea of which exons belong with which exon, or how frequently that is being selected, but I think it does a really nice job this. And it's the closest, if you ever do IGV and look for collapsed tracks, this is the closest to that. Okay, so we have one last thing we're going to cover. And so far for this gene region tracks, we've just been using this example data frame that's stored in the package Gfiz. But most of the time, you're not going to be caring about these 6 genes. You're going to be wanting to bring in your own annotation. And so that's super straightforward to do because Gfiz is built for Bioconductor. It talks to all the Bioconductor stuff, as we've seen with everything so far. And so we can use TXDB objects. And so we've been using the TXDB objects for everything we've done so far. The sequencing, not sequencing. We haven't been doing sequencing. The alignment, and then subsequent looking at the results, right? With things like feature annotation. So now we can use this same TXDB object to actually add to our plots that we're going to produce. And so in this case, we're going to use HG19. We're going to use the TXDB for it. So you'll notice a lot of our slides have HG19. That's just because HG38, it's not super new, but it's new enough that we haven't updated the slides because we know HG19 works, but HG38 will do exactly the same thing. And so we can do library, load it in. We have this TXDB object. And so all we have to do is use our TXDB object as in the gene region track function. It's super easy. So we use gene region track. In this case, we're specifying the chromosome. This will just save time. We load it in. And now we have a custom gene region track from our TXDB object. And so we can just easily reproduce gene models again from this TXDB. So we have a specific region we're focusing on, but we're just plotting tracks from it. And so we have these gene models here. And if you remember, I know it's a couple of weeks now, but when you played around with TXDB objects, we also learned how to make TXDB objects from GFFs. So it's very easy to get a GFF from anywhere. So a big source is the Ucsc. We can go onto here and go into their table browser. And so if there isn't a TXDB object for your genome, say you're working on non-traditional organism, a non-model, I don't know, like a zebrafinch or something like that, and there isn't a TXDB object, you can go to Ucsc, you can have a look. You can then have a look around, find the one that you want. So we're really interested in elephant sharks. Well, then we can find the GTF from here. Okay, and download it. Then once you've downloaded it, okay, you can then read it in. And so here we have an example with the HG19 again. Okay, and so we can read in this HG19 using make TXDB from GFF. Okay, so you give it where the GFF is, or a GTF, it will work the same way. Read it in, it's created a TXDB. Then you create a new custom from TXDB and you can plot it. Okay, and it looks pretty much the same, right? It just has a different annotation because of the different sources it came from. Okay, so there are some exercises. We did finish 10 minutes early. So I'm definitely open for any questions if anyone's been playing around with this. I also will be around for any questions if anyone wants to do the exercises, but I will not run through them. I know some of my colleagues have been going through and answering them. I just don't have time today to do that, I'm afraid. Sorry. Okay, so we're just going to go to the section 2. Okay, and we can go to slide. And we're ready to go. As always, I think it's nice you guys have been using the chat to ask questions and stuff like that. As always, keep doing that. Cool. So there's a bunch of stuff here at the beginning, which recaps. So links to the file formats in IGV and links to all of this. This is the beginning of the last session as well. That's the way you can download it all. Where the information is and where you need to be within your directory within R. So for most of us, because we're running these sessions back to back, none of this has changed and it's super easy to find. There's a quick recap. So if you remember, we've been using Gfiz and we created a series of different tracks. And then we can use the plot tracks to plot them out. So one of the tracks we created was an access track. We also imported some score coverage information using from a bigwig. And that's a data track. And so you can see we combine them with the C function and we can plot them all together. And so lastly, we had the gene region track. Okay, and we also showed the annotation track, but I think that is a broader class than the gene region track, which is the one we probably use the most. And so you can extract this straight from TXDB and then annotate a given region. And so hopefully this series of 3 shows you how you can build up to showing an actual annotated region of the genome that looks pretty nice, right? We have all the gene models. We have what we're actually seeing for this. I think it was a chip. I think it was a chip. No, it's probably, I can't remember what it is. I think it's a chip. I think it's a MIC chip. And we can also see the axis, so it's labeled properly. So this is a good spot to be in, but there's still more information and more tracks that we can add, which could either supplement this or be useful to illustrate different questions or different things. One of the key ones in this is a sequence track. Okay, so often you'll want to be able to show sequences. We've probably used this most if you're into any kind of, you want to display a mutant, or you have any kind of variant analysis, or if you have a motif that you care about. These are all reasons that you might want to show the sequence information, right? And so again, we can use bioconductor objects to do this. We can go to the BS Genome package, which we've used before when we did the bioconductor section. And you can just load that in because it's an R package, and you can easily subset it to specific regions, right? If you remember, we can use the double square brackets and a chromosome name, and that'll give you a DNA string with all of the sequence of that chromosome. And so once we've loaded that in, we can then just turn that into a sequence track. So you create it straight from the BS Genome object. And in this case, we're actually using a shorthand. So once it's once this is loaded in, there's a shorthand in the environment called H sapiens, which is just the sequence for Homo sapiens. You could use the full, you should be able to use the full length if you have multiple H sapiens loaded in. I think it takes this, will just go to default to whichever was loaded in last. So if you loaded in HD19 and HD38, if you did HD38 second, it would do HD38. But this is just laziness. But you can directly specify. But you just have to use the sequence track information function. And so now we have a new track. And so this is the S track. And just like before, we can use plot tracks. We can give it some coordinates and a chromosome. And then it will plot out the sequence. And it plots it out in this like traditional color-coded way. Okay. We can also control the size here. So we made it a little bit bigger using CEX like we did before. You can also specifically use a DNA string set object. Okay, so we can go to our H sapiens. We can extract out a string set from it and use that. And so this is important because if you're using, if you remember, we used the short read function, and that's used to read in FASTA files from outside of R. So you could read in a FASTA file from outside of R. It gets imported as a string set, and then you could use that directly with this sequence track. Okay, so we're just doing that, and it's pretty much the same way here. And we're just not doing the import. You can also skip that step. Okay, so this is an example if you're loading in your sample, loading in your FASTA and doing some stuff with it for another reason, and then you have this string set in your environment and you want to use it. If you're just doing it for graphing and you have the FASTA file, you can just load it directly from the FASTA file. So here we have an example. This is actually the same region. So all of these are the same region. This FASTA file is a FASTA file just from this region. And so we can just say plot the tracks. And this time we're specifying from 1 to 50 because that FASTA file starts at this point. So yeah, you can see there's a bunch of different ways to go from a variety of different sequence formats and get them into a nice sequence track, which you can then plot. And so there's also a bunch of ways to then play around with this sequence. There's a lot of information within a sequence, right, that can be useful, such as its complement. And so there's a argument that you can use for plot tracks, which is complement equals true. And so you can see that will just plot the complement of our original sequence. We can also add extra labels. So similar to what we did with the axis and also our annotations, you can ask to add 5.3 information onto it, or 3.5 if you prefer. And what that will do is it will add a 5' and 3' label to it. And it's smart, so it will know that when you do a complement that these labels get switched. Okay, so you can see 5', 3', now it's 3', 5'. And you can also control the size of the bases with CEX. So you've been seeing we've been doing that as we go anyway. When it gets to a point that any of you increase the size, and if any of the letters would actually overlap with each other, it changes its style format, because that would be horrible to look at. And so instead of doing it that way, what it does is it creates these blocks. Okay, and this is similar to IGV. So if you zoom all the way in IGV and look at the sequence, it shows you this, the actual letters. And as you zoom out, it becomes too much information for it to portray, because all the letters are right on each other. And so it resorts to this block format. And if you prefer it this way, like a lot of people actually prefer the visual representation as opposed to the letters. The letters it's a lot to process visually, and the blocks can be more informative. And so you can actually just force it with the argument no letters equals true. And so as you can see, these colors are we have this nice pairing of the dark red and the light red and the dark blue and the light blue. And so they're quite aesthetically pleasing, but there's also, there's the conventional colors for sequencing, right? The vibrant green and the yellow and stuff like that. So you can control the colors exactly how you want. And so you can control the colors to be that vibrant green and the vibrant yellow. Or if you're specifically interested in a subset of bases, say you're just really interested in Cs because you're interested in a specific kind of mutational switch and a transformation of the bases, then you can highlight the ones that you care about. And so in this case, we are creating a vector called col for letters. And we're saying the A is equal to dark gray. They're all equal to dark gray, apart from C, which we're saying is red. Okay, and so you can see the structure of this vector. We have the A equals, so it knows the equivalency between these, and we have it for each base. And so then when you do plot tracks, we're still using exactly the same arguments in the beginning, but we're just adding this new argument, font color. And so this is expecting a vector in this structure. And as a result, it looks like this. Okay, and so this way of making a vector is creating a named vector. Okay, so if you were to look at just col for letters, it would consist of dark gray, red, dark gray, dark gray. But each of those points would have a name, which is the first part. Okay. Yeah. Okay, so that's sequences. We went through that pretty rapidly. There isn't too much complexity in it, but it's a really nice way of being able to add, again, the sequence information onto your figure. Maybe you won't be looking at it at the same time as you're looking at these tracks and annotation files in quite the same way, but it's definitely a useful track to be able to add. And so, yeah, so far we really have this kind of summarized genomic information, which is often one of the most important things. We have all the associated metadata for genes, and that can be stuff that you've added. It could be ranges, it could be genomic features, it could be the sequence information. Another big reason that you might want to work with Gfiz is you actually want to show your BAM files. Okay, you want to show alignment objects, and then use that to be able to generate summary scores, like on the fly, without having to actually do a counting, for example, without having to generate a bigwig. And so to do that, we can use this new kind of track called alignments track. Okay, and again, we have a small BAM for you to try this out. Obviously, if you have a very large BAM, then that can be, it's a lot harder to work with, but you can always subset the BAM to regions that you care about before trying to load it in. Yeah, so we're just loading in this small BAM, and what we've created is, again, a reference alignments track or alignments track. And so, like there's this recurring theme. You create a track, it has whatever properties you want, and then you use plot tracks to actually visualize it. And so again, BAM files contain information. They're mapped reads, right? They have alignment, so they have not just position, but they'll have chromosome information. So you'll specify that in the chromosome parameter, just like we've been doing for the bigwigs. And so what you can see when we plot these tracks is you get all of the different alignment reads, and then you get a coverage plot at the top. And so this is a lot of information, right? Getting all of these aligned reads. And so it gets kind of squished a little bit. But depending on your sequencing depth, you'll be able to see all of these reads still. The other thing that's important to note is it's quite small, but hopefully you can see that, say for this example right here, right at the bottom, it has a slight point on one end. Okay, and we can also maybe see it clearer up here, this read that's by itself. And so the direction of the arrow is showing this strand of the aligned read. Okay, so you do still get the directionality as a feature of this. And so you can, as we saw before, control the type, just like with the bigwig plots that we did for data track. And so the options we've got here are pile up and coverage, which are included in the default. You can pick either or you can just use the default, which gives you both. And then there's other one called Sashimi. So if you were in the IGV session, you might recognize Sashimi, and we'll show you what Sashimi means a little bit later. And so if we specify pile up, we're just going to see this pile up without the coverage. Likewise, if you just want the coverage, you can get that. And so this is kind of nice because you can start to get this information without having to do any of the counting that we did to generate a bigwig, right? So this can be, if you have a specific gene of interest or something you care about, you could go straight from that BAM to look at this in like a couple steps. as like a first pass in with the data. Yeah, as before, you can use pass vectors into a lot of these arguments, and so we can use pile up and coverage again. And so I guess the key point here is the vector here does not display the order in the panels. And so we're asking for both a pile up and a coverage, but you have no control over the order here, unfortunately. However, this is definitely the right way to be visualizing this, right? It will look weird if it's the other way around. Okay, so Sashimi Plots. So we did cover this in the IGV session. If you weren't here, you can click on this link and then you can read more about IGV and how to use it. So the Sashimi Plots, they display the strength of signal spanning splicing junctions. So it's a really nice way of illustrating exon usage and which isoforms and transcripts are really present and which aren't. And so, yeah, we did this with some, in IGV, looking at some heart and liver data from the body map. And so we're just going to recreate this now using the same data, but we're going to do it in R. So both these BAMs are available to you in the data directory. And so we have to create two different alignment tracks for these. Okay, so we'll create a heart and a liver alignment track. This is also paired data, so you can set this as paired parameter to true. And then what we're going to do is we're going to plot them together. Okay, so we can combine these tracks super easily, remember, just by using the C function, combine function. So we're combining heart reads and liver reads, and we're combining it for this specific region on chromosome 12. And what you can hopefully see is with this, this is just the default build, right? And so we have this RNA expression data, we have this coverage at the top, and you can see it's quite nice. This essentially looks like you have these exons. And then in between, you have very few reads, but you can see the long lines which represent split reads. And so this is where the first half of the read maps to one exon, the second half maps to a second exon, and the in-between is been spliced out. And so it doesn't map contiguously to the reference genome. And that's represented here. And so if we ask for both a coverage and a Sashimi, what it will do is it will create these links representing the most common splice events. And so we're also going to ask for coverage for reference. And so you can see that, say, for example, we have this exon at the front. There's very strong appearance of splicing between these two. And we can see maybe there's slight differences here between our two tissue types, where it's slightly stronger, the choice for this one, while in this case, this choice is a little weaker. Okay, and you can just also see some, you can also see this change in splicing effect. This is really the key point, I guess. You can see that in this case, there's a very small splicing event. It occasionally happens some of the time, but most of the time it's here, but it's been changed to here in this data. And so this is a really nice way of displaying alternate splicing and changes in transcript and exonic usage. And that's not it for alignment tracks. There's a lot of other useful things you can do with it. And you can do that by controlling various different parameters. And so we'll go over some of the parameters. So we have col.gaps, col.mates, LTY gap, and LTY mates. Okay. And so for those of you who are in the plotting with R, you'll obviously, hopefully most people would recognize that col generally is a shorthand parameter for color, but LTY might be a little bit harder to remember. This stands for line type. And so this means you can start to customize the way these alignments plots look to help it better display where there's, say, a gap between, which represents an intron, right? So you have a split read, which is mapping over one exon and another exon, okay? And so in that case, we're highlighting that with red. The other thing you can do is label color the mate pairs. And so what we mean by a mate pair is we have paired end sequencing. So that means both pairs of the sequencing have been mapped to the genome. And so in this case, the gap in, the mate pair is colored in blue. So we can just look at the first read and you can see the reads here in blue, and then it's red is the gap between, and then it's blue. Okay. So this is a nice way to just, you have multiple forms of gaps in your data. And this is a nice way to color them to help differentiate between them. You can also play around with the line type as well, so that they have a different aesthetic value. And so in this case, we have these thicker dashes, and then a thinner dash in the middle, I believe. There's also line width as well, so LWD. Okay, so you've got multiple parameters over which to customize the exact aesthetic of your plot, which can be really nice if you're using these BAM tracks or visualizing BAM with these alignment tracks even. Okay, so really combining the two tracks sets that we just covered in this session, what you're often wanting to be able to do is show mismatches in the genome, and like look at SNPs and other polymorphisms. And so to really highlight those, we have to get combine both alignment tracks and sequence informations. And so to be able to do that, we need some information. We need to have like basically a reference to be able to compare to. And so then alignments, the alignment tracks can look at the reference and realize like, am I different or am I not? And so to be able to do that, we first create a sequence track based on the reference sequence. So in this case, we're doing sequence track and we're just doing the BS genome. Okay, so this data that we're looking at in the liver and which ones are we're looking at liver and we're looking at heart. So this is mouse data. And so then once you have this reference data in S track, in our sequence track, when you build your alignment track, you can provide a reference sequence. So we're doing the exact same alignment track function, same arguments, same input for the BAM. We've got is paired, but this time we're going to provide reference track. And so then when you plot these, okay, so we're going to plot our reads that we just made. And we're also, this is a annotation, similar to what we did in the last session. So this is from the txtb, it's an annotation file. You can see that the annotation is shown here, down here in yellow. But what's really important is it's focused. This is really focused in, and it's showing us mismatches. And they're highlighted in yellow. You can also see there's an additional mismatch here. We can then add the sequence track. So this first one, we built it with the genome information, the annotation, but we're right in the middle of a gene, so we don't really get much information from that. So we're going to switch over to a sequence track. And this can help us infer what's happened. So you can see in the reference there's a T, and in the observed alignments there's a bunch of Cs. And it actually shows in the coverage like what proportion of this region is Cs versus the standard. So you can see it's about 1/3 to 1/2. Likewise, you can also see here there's been a transition from a G to a T. But this is likely to be just a sequencing error, right? Because it doesn't have very much penetration in the throughout the alignments. So this is probably just a one-off sequencing error. And you can really see that up here. But this, depending on the experiment, right, is present in a large proportion of your sequence reads. So depending on what question you're asking, there's been a change at some point in your pool that you're sequencing. Okay, so that covers pretty much all of the visualization aspects of this. What we're going to talk about in the last third of this session is bringing in other data. Okay, so we've really covered the different kinds of tracks. If you remember, we have we'll build an axis track, then you'll build some annotation for it. You can also visualize some data. And then we just covered sequences and alignments. Okay, so these are most of the formats that you'll use. We've shown you how to put them together, and you can put all of them together in one go if you want. You can have every single type of data all in one plot just by simply doing building the tracks and then doing plot tracks with all of them in this vector. So you can really customize it as much as you want. So now we'll talk about getting some data from external sources. So we've been kind of doing that a little bit as we go, right? We've shown you how to drag in FASTA files. We've shown you how to interact with TXDB objects. But if you remember when we used IGV, you had the ability to drag all kinds of information from the server or websites and things like that. And so these like repositories, places like Ucsc and Biomart have a ton of information within them, and that's readily accessible to us through R. And we can use that to then inform some of the tracks that we build. And so to start with, we'll talk about Biomart and how we programmatically query Biomart. And so there's some functions inbuilt into Gfiz for doing this. There is specifically the Biomart gene region track function. So we can build a Biomart gene region track. So these are very specialist tracks that we can build. And so when we do this, we just say Biomart gene region track. We say what genome we want to build, the start and the end. We give it chromosome information. And we get where do we want to get it from. And so in this case, we want to get ensembles. Okay. And so Biomark Gene Region Track has lots of options for all of these. They have lots of different genome builds and consortiums that built these genomes. And so you can control depending on which genome build you want. This also gives you much more accessibility to genomes that perhaps aren't in Bioconductor, but are present elsewhere. So I think I showed you earlier for the GFF how to download it from UCSC. Or you could also use methods like this to programmatically go and do it without having to open a web browser. And so then when we do this, we have a Biomark Gene Region Track. We could actually just get a set of tracks that corresponds to the annotation for these regions, for this these gene models. Okay, and so it's quite straightforward, just a couple steps, and you've got all this genome information. You can also add your own filters. Okay, and so Biomart itself is a whole package that queries different places. And so when you establish a Biomart connection to, when I say establish a Biomart connection, it means setting up a connection between your R console and whichever server you're looking up. You can configure that quite a lot to control where it's looking. And so to get more information on Biomart, you can load in the Biomart function. If you look at list marts, this will show you all the different marts that are available to you. So I can actually, let's do that real quick. That's the one. Okay, so it's going to take a sec because it's going to load in all these different marts that are available. Oh, yeah. I don't know if you can see that, but that just killed my R. Yeah, that might actually be a bit much for it to display on screen. Let's try something else. Okay. Let me just bring that back. Okay, it's restarted. Okay, so I have a new plan. I'm going to save it like it's doing in the slides. So there's a reason we save it. So often when you crash things like that, this is a good lesson. It isn't because the process is too much. It's actually because you're trying to print the result of the process to your screen. And that is actually more consuming for it to do than to actually do the lookup. And so in that case, maybe we can use head on this, and it will show us those the beginning ones. Okay. So there's a bunch of different ensemble ones here, but you can look through this. Maybe we can try and be a bit braver. Look at the first 20 rows. Yeah. Okay. But yeah, so that shows you the marks that are available. In this case, we just want to look at ensemble, just the whole of ensemble. Okay, and when you do this, you create a mart. Within this mart, there's different data sets. Okay, and so we can list those data sets. You can have a look at which data sets you care about. And then you can then recreate the mart with that specific data set. So in this case, we're like, oh, we only really care about Homo sapien genes from ensemble. And so then we create MAR. Okay, and then we can look at further filters. And so you can keep iterating through this to subset your data. And so in this case, there's a specific kind of, I'm going to run this quickly so you can see the different filters. Now it's going to take ages. Okay, I'll leave that running and we'll come back to it. Okay, but it basically gives you, you can basically drill down in the database to very specific groups of genes or groups of annotations, whatever you're interested in. And so in this case, we want to care about this transcript set that have been called golden transcripts. Okay, so this is just a set of annotations that have both been annotated by Havana and Ensemble. So they're just like very high quality transcript annotations. And so you can then subset your data to just those by adding this filter. Okay, so we're back at the Biomart gene region track. We played around with Biomart for a little bit to figure out what filters we care about and what parameters to add. And then to use the filter, we supply a list. And we say we want the source to be Ensemble Havana. Okay. So this is basically the way to control for like versions and things like that. Like version control for annotations is really hard to manage. And so you just have to know exactly what you want. You can read around, look at what other people did. If you want to repeat what someone's done, you have to see what version they've used. And then you can use things like this to get the exact version that you need. And so once we do that, we retrieve our filtered gene models and we can plot them just like before. Okay, so we're just creating another track and then plotting them out. The other place you might want to look at is Ucsc. So that function we just covered was interacting with Biomart, then there's Ucsc. And so again, there's another specific track function for that. It's the Ucsc track constructor. And that allows you to query the Ucsc database instead. And so we really just have to give it what kind of track we're hoping to get from Ucsc. But there's a lot of data on Ucsc of a variety of different kinds. So this can be a really useful tool to just get some published data and have a look at what it looks like compared to your data. Okay, so first we use the, that should say browser session function and the genome function to, and this, all this is doing is establishing the connection, okay? This is similar to when we're using this, okay? This is establishing a connection. Okay, so we're saying we're starting a browser session and we want this session to be about HG19. You can use this function to see what's available. So again, this is similar to when we were doing listing off the different properties in Biomart, like list filters and things like that. And so we're going to be using the query. We'll say we're going to use this session that we just set up. Okay. We care about ensemble genes again. So we're going to compare between this way and the other way. And we care specifically about a G range. Okay, so we're going to make this G ranges for Ucsc genome. We want to care about HG19, chromosome 7, and between this region. And what we get back is a list of different options. Okay, so these options basically show us what Ucsc contains. What does this look up? When we look this up, what kind of things could we look up? So we could look up the ensemble gene, the ensemble peptide, like CDS information, where it came from in an ensemble to genome conversion, like all these different things. And so in this case, we just want to look up ensemble genes, keep it straightforward. We want to get a region track. We want to get information about the exon start and the end. And then we can specify where these columns, how these columns are named. Okay. And so the if you do # gene region track, this will give you the help for gene region track and help you see all of these different parameters and what they're doing. Okay, and how to use them yourself. But at the base level, we can just use these and that should work fine. And what we've done is we've created this Ucsc track. Okay. Both of these are essentially annotation tracks or gene region tracks. And so we can just plot them both. It's not actually, we can't actually see both of them right now because there's just not enough space. Do we have it here? No. So yeah, you can't actually see them because there's different space, but there is actually a slight difference between the two. If you go to our single page version of this, you'll be able to see the difference. But if you remember, when we did this, we specify this region. But in the other one, we didn't. And so the Biomart version of this has more genes on the outside. Just because we narrowed it in. But apart from that, despite the fact we're looking up in two different regions, two different places, we actually pretty much get the same thing back because they're both querying the ensemble database. Okay. And so these two tools are really, really useful because they enable you to just grab any kind of annotation that you want and then visualize it. So that's with annotations, but the really cool thing about Ucsc is there's just also a ton of data on the Ucsc. There's all the things like ENCODE and just published experiments are stored on Ucsc. And so you can get that information. They also have a lot of information about conservation on the Ucsc. And so here we're going to grab some data. We're going to get a data track instead. And we specifically want to get the conservation from this table here. Okay, so this is the phylogenetic table. And so with this Ucsc track, we're going to create a data track. It's a conservation style of data track. And so you can see this query broken down here. There's a lot of information going into here. It can seem like a lot. You can see we've also got controls for things like what the colors are, the limits, what type it's going to be. And so as before, you can directly control how the track itself is going to be visualized before you plot it. Okay. And so we can then plot this conservation track. And we're also going to plot some peak reads. So this is some CTCF peaks. Okay. Or CTCF chip signal. And so this is from a CTCF peak. And so you can see a nice summit in the CTCF here. We're quite a high resolution. We're looking at 250, well, 500 base pairs roughly in total. And And we also have a scale, a genome axis scale, but this is a scale as opposed to a full axis. And so we've plotted these three things, and you can. This is a really nice way of demonstrating in the top we have this conservation, and there's a peak in conservation right at where the CTCF is. So if you have a region of interest that you care about, right, you can pretty easily, once you figure out all these arguments, grab some extra additional information to show the evolutionary consequence of, say, just simple things like chip peaks, without having to do too much work. So hopefully this shows you the power of being able to drag in information from UCSC. Okay, so this is the end of this session, and it's the end, that's a wrap for us on Gfiz. And so what we have remaining is, I'll bring up the stuff again. Comparing data sets with PCA and heat maps, and then Metaplots, okay? And so these two sections, they're gonna cover a lot of this similar methodology, essentially, but initially for RNA-seq and then for epigenomic data. And it's really about taking your data, like what we've done so far, if you were joining us for either the RNA-seq, ChIP-seq, or ATACseq sessions. Once you have all this information, and you're able to interact with it in a fairly okay way, you've done some gene set enrichment, things like that. How do you handle the fact that you have all these different gene groups, and how do you simplify that analysis down so you can understand what's going on? And so we're going to be covering things like PCA, heat maps, and also Metaplots. Okay, so the next session we're going to be looking at comparing data across data sets. But I actually had a good question from King while we over the break, so I'll just quickly show it to you, just in case anyone else missed this. So when you're plotting these types of plots, this is actually something I struggled with as well, so that's why I'm highlighting it. When you plot these kind of plots, if these letters overlap, then it will form this, right? And this is working totally fine with this code to make the letters in my PowerPoint or my presentation here. That said, for you, when you're using your RStudio window, if you have depending on the plot size of your window, that will change the parameters of the plot output, which will change how much these will overlap with each other. So when you do this on your system, you might get this response instead. So it's just something to be aware of with your plotting defaults and also your plotting window. And when you do all this, you can also control it manually if you're using a markdown. I think when we covered our markdown, and if you weren't there for that, you can go back to our reproducibility sessions, which I'll show you in a sec. But you can control the figure width and height in the R markdown. And so that way, if, for example, you keep getting this as your output, but you wanted this, you can change the width of the figure that's coming out to make sure that you get this answer here. Okay. The alternative is you just use less sequence, right? And then it will have more space. Okay, so I also mentioned the reproducibility. If you go into here, this first session on reproducibility is all about markdown, so you can look at that. Okay, so we're going to go back to visualizing genomic data though, and we're going to specifically focus on part 3 now. So let's click on slides. Part 3, slides. Okay, so in this section, we're going to cover the concepts of dimensionality reduction, PCA and the loadings from PCA, clustering, k-means, and testing in gene clusters. Okay. So this is really, once you've done some RNA-seq, or if you have RNA-seq of 3 groups, this is really your next step quite often in looking at your data. Okay, and so just to remind you of the file types, you can look back here to look at some of the file types that we've met so far in IGV, and you can look back at all these different sessions which are relevant to what we're covering. Download materials are here, and you're going to want to orient yourself into the R course here for the code to work. And so far we've reviewed signal and annotation using the Genome browser and with IGV or with Gfiz. And quite often we want to, when we're visualizing, this is really great. You want to look at a specific locus, you have a gene that you care about, and so you can really focus on that. However, a lot of the time when we're looking at the, like say, RNA-seq or ChIP-seq or ATACseq, we want to evaluate what's changing over regions in a much more holistic way, a genome-wide scale view of our data. And that's not really possible when you're looking at this, right? You can see one position, you can look at the whole genome at once, and you can't really tell what's going on. So this is really becomes an issue because biological data is considered to be this very high dimensional data set because you're considering every single base pair. There's all these different base pairs, all these different genes, all these other enhancer regions that you care about. And so to be able to then tell like a holistic difference between what's happening in one versus the other when some's going up, some's going down, like there's not a clear often pattern. When you look at a growth scale. And so to be able to get at the patterns that are underlying this, we use things such as dimension reduction. So that reduces the amount of dimensions we look over, or clustering, followed by things like generating heat maps. And so the real idea of this, it makes the data simpler to both to interpret. And so it makes it much easier to identify patterns. And these patterns could be really important for your quality control, or they could be really important for deducting what's actually happened to your genomic data. And so in this session, we're going to be looking at some data we've looked at a couple of times. There's liver, heart, and kidney data set from Bing Ren's lab. It's adult mouse. And so this is obviously a slightly more complex experimental design than a lot of the time we've been following this one data set where we have activated and naive T cells. When we've been looking at RNA-seq, that's a data set we've leaned on. But this is more complicated. So we'll have to use some more advanced methods to be able to tease out what's going on. And so to start with, we'd use exactly the same approaches that we've used so far. We'd look at we do some counting in the BAMs, right? This is what we did in RNA-seq. So if anyone wasn't there for our RNA-seq session, you can go back and look at our RNA-seq session for exactly how to do this. But we've aligned some FOSS-Qs. We have BAM files. We're counting, and then we're going to use once we've done our counting, we have our BAM files, and then we're going to decide what to count in using a TXDB object. This is our database of transcripts, okay? And then we do the actual counting here with summarize overlap. So it looks at our BAMs and overlaps them with the exons, and says how many times do I have a read in each of these exons? And then we can follow this up by getting this into DESeq. So we just use DESeq dataset to make it into a DESeq dataset. And we can run the DESeq function on it. And then we can have differentials between these tissues. So in this case, we're asking pairwise differentials, right? Heart versus liver, heart versus kidney, liver versus kidney. So this is often the first pass of what we do. We'll get some pairwise interactions. And if for those of you who sequence with us, this is what you get as an output, right? From our pipeline. So if you do come for us for our pipeline analysis, you'll get a pairwise comparison between every group. And so what you want to be able to do next is actually start to analyze what are the changes that are happening across the whole data set. We're going to start to focus in on what are the patterns across the data set as a whole, as opposed to one versus the other. And so a good way to start to do this is to reduce your data set down to just the genes that matter. And so we covered this at the end of the RNA-seq course, but a really good way of doing this is doing an ANOVA-like statistical method. It's called an Lrt test. And what we're doing here is just looking for ignoring the groups model and saying, with our reduced model, is there variation across this gene? Okay, is there significant variation across this gene? And then if there is, we can then subset our data set to just those genes to care about. Another thing that's useful to do is to transform the data. And so quite often when you just look at counts, they're all on a very, it's on a scale which is non-linear. And so you have some very, very low counts and some very, very high counts, and then being able to look at those together in the same place is impossible. And so to handle this, what we quite often do is we use a function similar to norm transform. Okay, so what this will do is basically adds an artificial value because you can't log zeros. And so this is a pseudo count. And so you typically add 1 to everything and then do a log. And so norm transform function does this for us, and it does it on a DDS object. And we can get this DESeq transform object back. And so if you do that, you can then start to try and look at your data. And so in this case, we're just going to extract out the counts, and then we just do a box plot. Okay, and so there's a lot going on, and it's really, I find it, even at this level, it's very hard to tell what's going on. Unless you have very clear and obvious changes to genome-wide expression levels, this plot is going to look like this. And so if you're using approaches like this, what you might find is something that contributes to noise in your data set is smaller counts. Okay. And so what you can see here is this is from the library VSN. You can plot basically the standard deviation versus the expression level here. And you can see the standard deviation is really high and low data sets. And so it means smaller counts will generally have undue influence. So it means that the smaller counts are going to be more involved in your visualization and clustering than maybe they deserve to be because of their variance. And so an alternative to some of this norm transform options is to use rlog. Okay, so there's an inbuilt function into DESeq And this does a regularized log, which attempts to shrink the variance of genes based on their mean expression. Okay. So rlog count, you get the rlog of the counts. So here we have rlog of our DDS. This is what I use typically if I'm ever doing any next steps analysis. So if I'm ever making a heat map, I'm ever doing any clustering, or I'm doing some PCA. It's the rlog counts that go into this. And so you just have to use the rlog function on your DDS, and what this creates is a DESeq transform object as well. So it's similar to this. But if we look at the result here, so we have our rlog result, we put it into the mean ST plot, and then we look at this one. This was our original. You can see that this actually handles the variance a little bit better. So you can see the variance has shrunk. Even though the pattern looks pretty much the same, the variance has shrunk on the ST level. Okay, so the rlogs helps shrink that variance a little bit using like methods intrinsic to DESeq, which we covered in the RNA-seq course. Okay. So what we've talked about so far is getting your data ready to do the next steps. Okay. We've introduced this idea of reducing your data set down to a smaller number using the LRT method, and we'll show you exactly. We've performed the test, but we haven't done any of the subsetting yet. And we've shown you how to get some transformed count data that is suitable to go into clustering or into PCA or any of these other things. And so now we'll start to actually talk about some of these methodologies. And the first one we're going to talk about is dimension reduction. And so the idea of dimension reduction is you have thousands of genes that you care about over many, many samples, and then trying to find like an axis over which to compare these two then becomes tough because you're comparing over all these different genes. And so instead of trying to compare all these different genes, what dimension reduction does is it reduces it down to, say, some meta or eigen genes, which represent this kind of major pattern. And so it rotates the axis of orientation over which you're caring about your data so that really you're not looking at gene A in sample 1 versus sample 2, you're looking at an approximation of what most genes look like. And so by this, you hope that like the strongest patterns in your data will correlate with the with this axis that you're looking over. So principal component 1 will contain the majority of the variation, and hopefully most of the genes are doing the same thing within that group. And so, yeah, there's many different ways to do this. Principal components analysis is the main one. There's also other things in multifactorial scaling, non-negative matrix factorization. There's also, if you're into single cell, there's tsne and UMAP. So these are all useful methodologies to be able to dig into this. And so doing a PCA coupled with DESeq methodology that we've already learned is really, really easy because they have a function called plot PCA. And so you can take your, in this case, we're taking our log values, okay? Which is just, if you remember, we had our DESeq object, we did our log function on it, and now we just pass this into plot PCA. And the nice thing about this is if you're doing this, it remembers all of the different group information that you put into DESeq. So in this case, the group that we cared about was tissue. So we can give it as our int group. And then we can also control what parameter for the number of genes to be considered in the PCA. So obviously, well, by default, this only considers your top 500 genes. General rule is the larger genes or the more expressed genes have more of a role. But if we're doing this variance shrinking by getting the R-log values, that becomes less of a, you don't need to limit it to 500 as much. And so in this case, we're gonna do it for the whole data set. So we do n-row R-log tissue. Okay. And so this means we're going to use the whole of our data set in the PCA. And so then when we do this, we get this back. And so this is our PCA plot. In PC1, I think actually if we go this, yeah, this will explain it. So the PCA shows the separation of your samples in this two-dimensional space. There are many more components, but it only shows the first two. And what these are just comparisons along this imaginary axis that we've derived to show how these data points separate. Okay, and so the nice thing about the Pca plot built into DESeq is it tells you how much variance is attributed to this axis. So along the PC1, 51% of the variance seen in your data set is in that PC, while along PC2, there's 44%. So considering these two axes, that accounts for 95% of the variance across your data. And so looking at PC1, it seems to be very much be, if we just look at it in this way, PC1 explains the difference between all three samples in a graded way. So heart is all the way negative, kidneys in the middle, and liver's all the way positive. Okay, so this would suggest that kidney is, for at least for the components that contribute to PC1, kidney is almost like an intermediate between heart and liver. Then if we look at PC2, heart and liver are exactly the same within these genes, but kidney is completely different. So 44% of the variance is accounted for by differences between kidney and heart and liver. So this is an interesting and useful pattern to then start to be able to interpret. And so PCA is often used to be able to start to get a handle on sample similarity. One of the things I didn't point out there is that we have replicates in each, and each of these clustered together very strongly. So this is a really nice sign. This is, if you got data that looked like this, you'd be very happy, because it means the majority of the variance you're seeing is between sample, between group variation, and your biological replicates show very little variation between them. So the majority of the your results, and the majority of what you're going to be looking at, is biologically relevant variance. So as I mentioned, this is a super easy function, right? Plot PCA, you just add a couple of things and you get this nice plot that summarizes everything. This is super great. Sometimes you want to be able to do the PCA yourself. When you do the PCA yourself without using this special function that DESeq has, it gives you more control over the DESeq, over the PCA. And so to be able to do that yourself, you use the PrComp, principal components function, PrComp. And then you're going to be able to get much more information from the PCA. And so the input for this has to be a transposition of your matrix. So we have our R-log matrix that we grabbed. And so you have to transpose this. The reason is it's just expecting the information for you to care about to be ordered in a specific way, i.e. the separation between the groups that you want to separate out, say between liver 1 and liver 2, it expects those on the rows instead of the columns. So you just have to use the T and it spins it round. So the columns become rows and the rows become columns. And when you do this, you can save it and you now have this object, which is our PR comp special object. And in the X slot of the PR comp is the mappings. Okay, and so what do we mean by mappings? This is basically the information that goes into your graph. So you can see here on the PC1, we have minus 100, minus 120. Okay, so this would correspond to these two and their PC1 scores. Okay, likewise, it has their PC2 scores. But it doesn't just have these, it has all the PCs. So if you wanted to hunt down the missing 5%, you could look in some of the PC3, PC4. For a data set like this is less important, but quite often your data won't look as pretty as this. This is a very, very tight data set. More common than not, I might see 40% in PC1, 20% in PC2, and then you've got 40% still left in the remaining principal components. So you might want to look further deep down, especially when you look at things like chips, right? When you look at chips, you're often using an input, and the input is normally the difference between inputs and chips are normally the massive there's massive variation there. And so that automatically rules out your top component, and you're then really interested in PC2 and PC3 as opposed to PC1. Okay. And so because this information is for the graph is all here, you can then just recreate your graph really simply using base plots. So we're just saying we want to use X, and we're going to use the colors based on the tissue information, and then we add some legend as well. And so this is the same plot, same information, but you've recreated it yourself. So once you've made a PCA, especially if you have something nice that looks like this, the first question always that comes up that I ask, that I get asked by other people, is what are the genes that are driving this? Okay, so we know that this abstract rotation that's happened, and we're comparing over this reduced dimension, but like which are the genes that are mostly responsible for the difference, say, between heart, kidney, and liver? Or what are the genes that make kidney so special? And so to get those out, these are called the rotation or the loadings for all of the genes. And so they can be found for every single PC, every single gene in the rotation slot. Okay, so you can go PC res rotation. And so the genes with the highest score, the highest values, or the extreme lowest values are the ones that are really driving this. And so say a gene with a very, very negative value will be driving towards heart, while a gene with a very positive value will be driving towards liver. And so the label on the row axis will be whatever label you put in for the input as well. And so this is a really, really nice way of being able to then start to figure out what's going on with our groups. And so in this example, we're going to investigate what samples along the negative axis of PC2 are kidney specific, what's driving that kidney specific pattern. And so you can just look at the genes which are most negatively contribute to PC2. And so in this case, we're just extracting out PC2, and then we're sorting it by decreasing, and we're extracting out the top 100. And actually, we're just going to look at the top 10. So this means this gene right here, 20,505, is the main one for this. And so I haven't actually checked this, but we could go to NCBI. And we could go look at gene and have a quick look. I'm hoping that this is something really kidney specific. That looks pretty kidney specific. Solute carrier family, sodium phosphate. That sounds like something the kidney would care about. And if you scroll down to the mouse encode transcriptome data, it's expressed in the kidney and nowhere else. So that would explain that, right? That would explain what's specific about the kidney. So this is a really nice example. So using this methodology, you can start to subset your genes down into genes you care about, not using a pairwise method, but using your whole data set at once. And so what we can do now is use your, say we're considering these top 100 to be our PC2 markers. We can then subset our pairwise comparisons to have a look what is special about these pairwise comparisons. And we could then plot these, and you can see here, right? These are, again, I looked at just one. But in this case, we're looking at every single pairwise. And so if you look at these genes in heart versus liver, and the log2 fold change, not much is happening. If you look at them heart versus kidney or liver versus kidney, their kidney is actually upregulated. Wow, there's two ways to interpret this. Either the heart and liver are both downregulated or the kidney is upregulated, right? But you'll be able to look at the actual counts as well if you wanted to confirm which one of these is happening. Okay, so that's just all with subsetting based on this PC2 information. Okay, so that's handling your PCs. And you can start to see how you can quickly get gene sets that you care about in a totally different way. In the previous way, we always talked about you get the adjusted p-values, right? And then you look at maybe which ones are upregulated or which ones are upregulated twofold. And that's how you generate a gene set. This is leveraging your whole data set to be able to get a gene set which you can then look at more in a more focused way. Something else you might want to do is start to do some nice level of control and comparison between data sets, is sample to sample correlation. So this can really support the work you do with your PCA, which is showing this nice tight clusters of replicates and quite differences between your biological samples. And so to look at this, we can use the core function. And what this does, if you have this matrix, is it will assess the correlation between each of these samples. Okay, and so you can see obviously there's a very tight 1-to-1 ratio between the same when it compares against itself. But then if you compare like heart 1 to heart 2, it's very high correlation, and then that drops off with other tissues. Once you've made this is a matrix, right? And so what we can do is take this value and and convert it into a distance measure. And so distance measures basically give you an idea of a dissimilarity. And so the way we do this is we do 1 minus the correlation score. And then we do distance. We turn that result into a matrix. And then we can put it into a heat map. Okay. And when we do the basics with pHeatmap, okay, so this is, I think this might be the first time we talk about pHeatmap. pHeatmap is awesome. It's used for making heat maps. So we use the pHeatmap function. You give it a matrix, which is our distance matrix. Okay, so we've gone through these various steps to turn our correlation into a distance object. And what it will do is then use these sample distances to do some clustering. Okay, and so this means it doesn't matter what order you put these in, it will order them in a way where the closest ones to each other are put next to each other. And you can see the tree on the side. And so it seems that kidney and liver are more similar. And then heart is the outgroup. Yeah, and the nice reason for doing stuff like this is it really hammers home that your replicates look very similar. Okay, they have a very, very low level of dissimilarity. And because this is really this scale, it doesn't need to be bimodal. It doesn't need to have 2, 3 colors in it. The common thing we'd do is then apply our own gradient. And so we can use our color brewer, which we met in Ggplot. We're going to say we want 9 colors in blues. Sorry. And we generate these colors. So this creates 255 colors in this gradient. And then we can supply these colors to the plot. Okay, so this is just showing you what this axis would look like. But really, we're going to supply it to the colors to our color argument in pHeatmap. Okay, and so you can see it's a nice dark color down here, and then it gets lighter when there's more dissimilarity. So this plotting function here is just in case you want to play around with the gradient and actually visualize what it looks like before you do pHeatmap. Okay, and then the final level that you might want to add on top is add in some annotation information. So at the moment we have labels for each sample, but you might just want to have some, like here we have annotated our tissue. And so you can provide this as a little data frame. And it will go into that. This is a super easy thing to do because we have a lot of the metadata already in our call data, in our DDS object. So we can just extract that using call data. We turn it into a data frame, and then it's ready to go. And so then you can supply that into the annotation column, and then you get these extra pieces of annotation here. Okay, you could also do annotation row if you prefer, but or if you want to do both, you can, but it's not strictly necessary because they represent the same. Okay, so in the final 20 minutes, we're going to overview clustering. And so this shows you some early signs of clustering, right, where we're clustering whole samples. But quite often we want to actually cluster on a gene per gene level. And so this is another way to extract out patterns from your data set so you can get genes to focus on. Okay, so there's a lot, like we've gone over, there's lots of different ways to do this. If you have pairwise, you can look at the DE-seq results. You can look at PCA like we did earlier and we got the loadings, or you can do clustering. And so when you have really complex data sets, this can be a really nice approach. And it's one of the most common things I do with data analysis. And so to start with, what I'll do is I will quite often subset what the raw materials you need for your clustering is the rlog counts, right? And so that's what we've been using so far. But for clustering, it's quite a computationally intensive procedure. And so instead of using all of the counts, what we can do is we can just use the ones that we know are significantly changing at some point. And so this is where the Lrt test comes in. So in our Lrt test, we can filter down to whatever value we have here. So in this case, we're going to say we want to filter all the genes that have a adjusted p-value of 0.01. And they're also not NA. Okay, and we're going to just extract out the row names for these. Then we're going to subset our matrix based on these. Okay, so we're going to get the row names of our matrix and ask if they're in these significant changes, and subset our log matrix based on that. So now we have a matrix and it's 8,000 big. And so typically with these matrices, I don't think we showed you the value of it before, but this should be based on pretty much all genes. So we're talking tens of thousands of genes as opposed to sub 10,000. We might have somewhere between 10 and 20,000 before it. So we filtered about half of them out or something like that. So now it's much easier to work with computationally. And so again, we're going to turn back to pheatmap, and pheatmap has the ability to cluster inbuilt into it. And so by default, it's going to do some hierarchical clustering. And so what we do is we provide our significance matrix. We ask for scale by row. What scale by row means is instead of showing the raw count value, it scales based on, it scales each row using a z-score. And so this allows you to see the trends in gene expression across the data set. So even if, say, this gene here at the top, we've got a gene here, for example, maybe this one's expressed really, really highly, and then lower down here, we have something that's expressed pretty lowly. They're clustered together because they have the same pattern of they're low in heart, high in kidney, low in liver. And then they're visualized in much the same way, as long as the fold change looks kind of the same. And so that's what goes into the z-score. And so it uses this hierarchical clustering to then gather them together. Okay. And so what hierarchical clustering is doing is it starts, it considers your whole data set, and then it says, if I divide it in two, which division into two can I get two groups that are most similar together? And it is a style of clustering which is considered to be divisive, where it just gradually separates everything down. Okay. And so you can then like look at this tree and see which are most similar together based on the tree. So you've got like this group up here, you've got probably another group here, and then maybe another group here, maybe dividing this group into two. Okay, and you can cut this wherever you want. Oh, that's the same, it's a little bigger, so maybe it's easier to see. But you can see there's clear patterns that you maybe want to dissect out. Okay, And so this is hierarchical clustering that's gone into this. And pheatmap also does something called k-means clustering. There's lots of different ways to cluster groups, okay? And they all have different pros and cons. I'm a fan of doing both hierarchical and k-means and then having a look to see if they look kind of the same. I think that's always a good strategy when you have multiple ways of doing things. And so with pheatmap, all you have to do to get it to do k-means clustering instead is tell it how many clusters you want. And you provide the same pheatmap information, scale by row, sigmap, and it will do the clustering and it will actually divide them up for you as well. So we're actually going to save this out into K. So it's important that when you want to do clustering and you want to get the cluster information back, you save it out afterwards. When you do it like this, what it actually does is it breaks each of these down into groups. And so you can actually see what each cluster, the pattern of each cluster. Okay. And so here we have clusters, 1 through 7, and like 1 and 3 are kind of similar, right? 6 and 5 are kind of similar. 4 and 7 are kind of similar. But you can start to see there is slightly different patterns between all of them. And so the reason that you need to save this out is you can then extract the cluster information from this object. It doesn't just contain the heat map, it contains cluster information. So if you go into K, you use the $sign to access it and go into k-means, you'll get all the information to do with the k-means clustering that was performed. And so in cluster, it's a simple data frame containing the gene and what cluster it belongs to. So you can extract that information from here. We're turning it into a factor, then a data frame. And then we can have a look at it, right? And you can see we have all the cluster information. And because it's now a data frame, you can then go back, draw your pheatmap, right? And then look at the clustering. Okay. And so actually, what we're doing here is, sorry, I didn't explain all the steps. What we're doing is we're first doing an order cluster data frame by cluster. Okay, so what this means is that it's ordering our significance matrix now by what cluster it belongs to. And then we can do pheatmap on that ordered cluster. And at this point, we turn off the clustering within rows because we already are providing clustering information. If you had clustering in rows, it would still do the hierarchical clustering, but then you're labeling k-means clustering and it will get confused. It will look weird. Okay, we're also adding in this the cluster information as the annotation. Okay, so this is the result. And so there's no clustering on this side that's being performed, but we're ordering by the clusters. Hopefully that isn't too confusing. But the pheatmap itself, while we're plotting in this specific function right here, is not doing clustering on a row basis. But in our previous pheatmap here, we figured out what the cluster of each gene. And so then we can label them. And we've ordered our matrix by this. Okay. And so you can then look at the pattern in a gene by gene way. Okay. And so one common question when people start to do clustering is, how do I know how many clusters to do? We kind of took a run and a jump and guessed that 7 would be appropriate. And so there's lots of different ways to do that. Too many to go into detail in all of them, but a classic way, especially with k-means that people use very commonly, is something called the silhouette method. And so what the silhouette does is it evaluates the similarity between each cluster member to the similarity between the clusters. Okay. And so here's the math for it, but basically you're looking at the dissimilarity of a cluster member to its own cluster, and then that's calculated as the average distance between cluster members and all other members of the cluster. So really what this is trying to do is minimize intra-cluster variation and maximize inter-cluster variation, so that your clusters and the genes within your clusters are very like each other, but very different from other clusters. Sorry, I'm getting to the point where I can't say cluster anymore because it's getting a little much. And so to calculate the silhouette score, we can use the NBclust package. And what that will do is it will calculate it for various numbers of clusters, and then you can see which is the best. So it's basically going to do the clustering a bunch of times, assess which is the best clustering based on this metric, and then tell you which one you should use. And so to do this, we just supply our significance matrix. What you'd also want to do, which is the optimal strategy for clustering most of the time, is to scale your data. And so if you remember when we looked at this, right, this is scaled by a z-score. And so we can manually do the scaling ourselves using the scale function. This means the patterns in like whether a gene is expressed really highly or really lowly isn't driving the clustering, and instead it's the pattern, because it's the pattern we care about. We don't care about which genes express the most. And so we do scale, and we're doing this T transformation because the scale function likes the matrix in one orientation, so we have to turn it into that orientation. But NBclust likes it in the original orientation, so we turn it, we do scale, then we turn it back. And then the last couple parameters I'll show you is min NC and max NC. So this is the min and maximum number of clusters. Okay, so you can control how many clusters you look over. Like you don't want to look at one cluster because that's just your whole data set. Right? That doesn't matter. You might find that you want more than 2. And so at the moment, this is set up, it's going to look at between 2 and 12 clusters, but you can dial that in depending on how many groups you think is reasonable based on what the heat map looks like. And so after this result, we find out that the optimal number of clusters is 3. Okay, and that's stored in the best.nc slot in the result. Okay, and so if you go back and look at this, it might seem that you can get more clusters out of this, and that's totally fine. You don't have to go with the optimal number, depending on what questions you're asking. But the most parsimonious and the one that has the most separation between groups is 3. And so once we've done that, we can then extract out the cluster information for the best partition. So it saves that information so you don't have to do the clustering again. And so if you look in best partition, it stores the number of clusters, or which cluster each gene belongs to, we can then sort based on this if we want, our significance matrix, and then we can have a look. And what we actually did was we included as the annotation our old clustering, okay? And you can see that broadly, this cluster, right, is pink and purple. This one is the greens and the red. And this one is like the teal and a little bit of some of the other ones. And it basically divides it into 3 clusters of, this is when one tissue is up, this is when the other tissue is up, and this is when the other tissue is up. Okay? So it's really simple to interpret. And so this is just another way to derive a gene set, right? We've now got a clusters. This is a group of genes we care about. And when we have a group of genes that we care about, it's a gene set. That means we can do functional testing in exactly the same way as we've done before. For those who missed it, we covered a lot about gene testing in the RNA-seq course. So you can go back for more detail there. But I'll run you through a quick example here. So in this case, we're just going to ask which ones are in cluster 1. These appear to be heart-specific genes, okay? Yeah, this is cluster 1, I think. Let me check. Yeah. Okay, so we'll just get the row names. We'll ask, first subset, which ones are equal to cluster 1, and then we'll extract the row names of these. Now we have the row names for all of the cluster 1. We can then generate a logical vector for GoSeq. So as our background, we want every single gene that is changing. Okay, so that means every gene that isn't an NA for the adjusted p-value. And then we'll find out the heart-specific ones by asking which of the background are in the heart-specific. So that will give us a true-false, right? And so we can then add the names to this, true-false. And then now that we have a logical true-false, we can pass it to GoSeq. So we load in GoSeq, we ask it to tell the null distribution. And so what the null distribution is making sure there's no bias in gene length for Go enrichment. And so if you do this step, it will normalize out the bias towards any biases seen in gene length. So there is some bias here because it's not a straight. And so we'll run null p. We have it on our heart specific. You can also, you give it this information as well, what genome and where this genome has come from. And then the result of this to test, we can then chuck in the actual GoSeq equation. Okay. And so we're going to test against biological processes for Go. And we run GoSeq, and the top terms coming out are stuff like muscle structure development, circulatory system development. These are all unsurprising and very good terms to be coming up for heart, right? So we've subset to our up in heart data sets or genes, and it looks like we're getting back the right terms. And then we can also look back at the PCs now with this information as well. We can use our heat maps to understand what's driving PC1. And so what we can do is sort our heat map based on our expression matrix based on the loadings and its effect on PC1. Okay, so what we do here is we're extracting out PC1 we have the result of the PCA, get the rotation. So that's the per gene information for the PC1 or for the PCs. And then we're extracting out specifically one. And then we want to sort it by decreasing equals true. Then we can use the function match. And what match does is it asks, we'll look at the names in this and the row names in this. And it will return the numeric of where the match happens. And so this allows you to reorder one vector based on another vector. Or in this case, reorder one matrix based on another, on the order of something else. So we're using the row names here and the names here. We see which ones match, and then we reorganize our matrix based on this matching. And so now the matrix is ordered by the PC1 rank. And so then we have this PC1 matrix. We can then put this into pheatmap. Again, we'll turn off the cluster rows because we're providing our own sorting for the row. And if you remember, we're looking across PC1. So if we just look at PC1, we have a heart, then we have kidney, then we have liver, okay? And then if we look at the heat map, we have heart, and then kidney, and then liver. Okay, and so kidney does seem to be an intermediate between heart and liver here, which seem to have opposing gene expression patterns. Okay, so that is dissecting RNA-seq data using PCA, using clustering, and using some of these heat map visualization approaches to really get an understanding of some of this much more complicated data sets. These skill sets are incredibly useful, and you can spend hours separating your data out in hundreds of different ways. But this is where the bulk of my time with analysis often goes. It's splitting the data down into different ways and trying to see what patterns look interesting. And then you can start to ask what's driving these patterns. Okay. And this is just scratching the surface, but it's a good jump off point for anyone who starts to work with data sets like this. Okay. So we have one more section. We're nearly there. Okay, so the final section is we're going to be taking some of the things that we've learned already, applying that to ChIP-seq data, and then bringing in some new tools as well, because ChIP is special. Along with ATAC, anything epigenomic, you care about slightly different things. And so there's other ways to start to pass this information and to visualize it in a way that is both informative and aesthetically pleasing. Okay, so we're going to get back to that in half an hour at 3:30. Okay, so we've got one more session, and this session is going to be focused on visualizations that are specific to epigenomic data. Okay, so this is going to be things like ChIP-seq, ATACseq, MNA-seq, all of that family. Okay, and there will be, yeah, so that's kind of the focus. There's been this slide in all of our presentations, but you can look back to how we've looked at file formats and IGV before. Also link back to all of our different materials here. And then here's the download and where to get to in our course when you set your working directory within R once you've downloaded the material. Okay. And so what we did before is we've looked at, we talked about IGV, but like really how we did this with Gfiz is the really big thing. We spent this morning talking about extracting information and then displaying it as a track. And then we also talked about how with RNA-seq data and ChIP-seq and and things like this, we can look at a single locus like this, but really this data set is highly dimensional and there's ways to reduce that down. And we talked about how to do that with RNA-seq. So we looked at some PCA and we did some heat maps and some clustering, which is great. And we're really focused on how to do these for RNA-seq. Most of these methodologies that we just went over, there are methods to do it with epigenomics data. Plus, there's also additional ways that you can present your data through things like these metaplots and heat maps, stacked heat maps. So we'll talk a little bit about doing some of the things that we covered in RNA-seq, going over that again so that it really makes sense. And then we'll talk about some of this metaplot stuff. Okay, so a lot of what we'll focus on for the metaplot analysis really is a package called Soggy, which was developed by Tom. And we'll also talk a little bit about Profile Player. But there are other options within R. There's things just general heat maps, enrichment map packages, NGS plot. And then outside of R, there's Homer, and a really big one that people may have heard of is Deep Tools. Okay, so Deep Tools really pioneered the use of these, made it easy the use of these stacked heat maps. And Doug from our team has taken that methodology and adapted it into ways to use it in R. So we'll touch on that in the end. Okay, so in this session, we're going to be focusing on some ChIP-seq data, but you can think about this in much the same way if you had ATACseq data or any other kind. In our ChIP session, we focused on a MYC transcription factor. In this case, we're going to focus on CTCF. Okay, we're doing that just so we have something different to look at. CTCF is also a very, very strong chip. So when we come later to doing metaplots, I'm thinking about a little bit about motifs. It's a very good data set for that. So just to kind of recap what the position the data's in, this is kind of the tail end of, let's say, the ChIP-seq approach. Maybe you've generated some peaks already. We have some peaks in this data folder. We can use dir, the directory function, to get the names of all these peak files. Then we can loop over them with a for loop and read each of them in, and then turn them into a G range. Okay, this is being assigned into a list. And because the list is made up of G ranges, we can then turn that into a G ranges list. So I know this is not the most intuitive thing, that if you have a list and it's made of G ranges, why does you have to do an extra step? When it's specifically a G ranges list, it's a specific class of list, and that has inherent properties which gives you more functionality. So this is a useful step to do, because then it knows that the whole list is a G ranges, essentially. We can add the names. And once you have that, you can then start to identify what we consider non-redundant sets of peaks from this. Okay, so this is a common step in any kind of peak-based methodology. You're going to have a couple replicates of each type, and you want to make sure that the peaks that you care about are present in the majority of your replicates. So if you only have 2 reps, they want to be in both. If you have 3, you want to get 2 out of 3. That's the kind of ratio you're aiming for. And so that's what we're doing here. We use the reduce function. And what reduce does is it gives you a list of every single potential peak. Okay, and so if there's anything contiguous that are overlapping, it will be reduced down into a single range. Okay. And then once you've done that, you can ask back in your peak set, do any of them overlap with this? Okay, and so this defines your non-redundant peaks, and this asks which data sets have these non-redundant peaks. And if you go through these steps, you will end up with a logical matrix saying true or false, are my peaks present or not present? Okay. So if you remember, all we're doing here is we're generating a list through asking that question. We're binding that list together into a data frame. We're adding the names, and then we add that to the non-redundant peak gene list, gene ranges. So that's what we end up with here. So true, false, is it present? And so once you have the true, false of whether it's present or not, in our case, we have 2 reps. So we want to make sure that every single peak is present in at least 2 of its biological conditions. Okay, so for a peak to be a true high confidence peak, it has to be present in both CH12. So this guy looks good, or it has to be in both MEL data sets. It can be present in both, so it's present in all cases, but it has to be at least 2 or 2. Okay, and so we do that here. We're asking, is CH12 mcols greater than 2? Okay, and we're applying row sums. So when you do row sums on anything that's a logical, every true becomes a 1. Okay, so we've done this. We've filtered it down to our high confidence peaks. And then once you have your high confidence peaks, then you can start doing differentials. So we get our SAM tools, we have our genomic alignments. We can then say where the BAM files that are associated with these peaks are. We create a BAM file list. And then we can summarize overlaps over this. So that means we're counting how many reads are in each of these high confidence peaks. Okay, and this can take a while, this step. And it's all controlled, the memory is controlled by this yield size, if we remember. So we're reading in 5 million reads at once. You can tune this down if that's killing your computer. Okay. And so once you have all this, you have your counts, you're all good. Next, you can make a DESeq dataset from your counts and then do differential expression analysis comparing MEL to CH12. So we get some results. We want them as G ranges. Okay, so at this point you have a nice pairwise log2 fold change between these samples. You've done the majority of the analysis that we covered in the ChIP-seq pipeline. And this is the kind of data we give you at the end of the pipeline that we do if you do some ChIP-seq analysis with us, with our basic pipeline. So that's great, but now we're into the next steps, okay? And so as you remember from the session before this, we want some counts to start to interrogate things like PCA or to interrogate clustering, things like that. And so the place we start is by doing the rlog of our DESeq data object. Okay, and so if we remember, this squashes some of the variance associated with low counts, and it regularizes it, so that means all the samples are comparable to each other now. And so actually this heat map looks a bit too big to be right here. But what we can do is we can do a similar correlation matrix between samples. Well, you can see enough of it to know what the result will be. But if you remember, what we just did is we do the correlation between the counts using the core function. Perfect one-to-one correlation gives you a value of 1. So if we do 1 minus this essentially reverses that score. And so we can then use it as a dissimilarity metric. And we can get the distance. We're using as.dist. And then we can feed this into the p-heat map, and we can determine the clustering based on this distance. Okay, and you can see that there's a high level of similarity, and it's tissue cell type specific. Okay, and the CH12 is down here, but you can see that both 1 and 2 are represented here by this cutoff. Sorry, it seems like some of the graphs are rendering a little bit weird. So apologies for that. Maybe if let me try something, see if it's better in this single page. Okay, so actually this is a good point for maybe some of you who are just joining us now, or yeah, who have forgotten. A lot of these plots, this one still looks weird, but you can see this one. Everything is also present. Everything that we produce is present in a couple formats. We have a single page format and a slides. Some people find the single page easier to work on because it's in a nice scrollable document. You can just keep scrolling down through. But we'll go back to the PowerPoint or the presentation. I always say PowerPoint. That's proprietary and not what this is. This is just a presentation of slides. What we can do here is you can then use the plot PCA function, just like we did before. This is rlog going in. And what we can see is, this might be the reason it looks funky. So PC2 has 0% variation, and 100% of the variation is between CH12 and MEL. Okay. So that means there's incredibly little variation between the replicates that is coherent and along a different axis to the separation between CH12 and MEL. And this is considering the top 500 most variable sites. Okay. If we were to instead, okay, so we didn't give any information into the n-top, so it defaults to top 500. We can give more information. So we can say n-top, and we say we're going to use the whole data set. So we're going to use nrow function on the rlog object. So it's going to figure out how many rows it has. And so we'll consider everything. And so now there's 96% variation in PC1, 2% in PC2. So that means there's another 2% in some of the other principal components. Either way, we have really nice separation between our samples. And we have a component that seems to have all the biological variation. So this is cool. And so we can do something similar to what we did before. But I'll show you a nice little trick. If you do plot PCA, what you can do is directly save this object. And similar to PR comp, if you've included the return data, instead of making a plot, it will give you the data. Okay, so in this case, it gives you the PC values. So this is nice because then you can plot additional metrics on this. And one of the big things that, if I'm ever concerned about a ChIP-seq experiment or even an ATACseq experiment, is I would add the fragment length. Okay, so this is a proxy quite often of basically, is there differences in sonication and or chip efficiency between my samples. Okay, and obviously if you're doing something biologically meaningful, you want the experimental conditions to be identical. So you can add this additional information in using Ggplot. So by extracting this information here, right, from PC1 and PC2, you can then draw it with a Ggplot. And we're just manually adding in the fragment length information here. Okay. So you'd be able to deduce what the fragment length information is using the ChipQC workflow, which is covered in the ChipSeq pipeline. And so if you, again, weren't here for that, you can head back to our ChipSeq sessions and have a look at how to get things like fragment length. Okay, but for now, we're just going to add them in manually like this. And then when you do the ggplot, we can say, I just want color by group, but I'm actually going to change the size of the dot to represent the fragment length. And now we can look at all of these things on one plot. And so you can see that there's actually quite a high degree of variability between the MEL samples. Okay, well, these samples are roughly consistent. So these three look pretty good, and this one is out on its own. That said, looking at the pattern, the divisions in the PCA is not driven by the fragment length, okay? So I recently had an experiment I was working with someone with, and we looked at the fragment length, and we also looked at the reads in the peaks, another sign of potential chip efficiency issues. And we saw PC1, we had a separation between our inputs and our chips. And then PC2 was a perfect correlation between PC2 and our fragment length, and also the reads and peaks, okay? And then PC3 is where our actual interesting data was. So we took a first pass, we looked at PC3, we used that to try and find genes that we cared about. And then we did some much more advanced analysis for batch correction using the knowledge we had at that point to fix that. And so there are ways to deal with things like this, but really this is a really nice way of being able to start to look at some of these QC metrics and see if they confound any of the observations you see. Okay, so we can go back to doing our comp function, principal components function. So we can actually start to extract out regions and peaks that are actually driving this separation. And so we do, if you remember, we take our log values, we do assay to get out the counts, we do T because PR comp likes things rotated. If you didn't do T, just as a heads up, what would happen is you'd have a dot for every single gene. And you'd probably kill your computer with it trying to plot it. So that's just a heads up. Then what we're doing here is extracting out the rotation along the principal components 1. So this is the loadings, and we're ordering by that. And so then we're ordering specifically the row names. Okay, and so that means these first 3 regions here are the regions that are the most driving the separation, and the ones that are really the ones that are changing the most, or driving the pattern and differences between MEL and CH12. And if we were to look at them, you can see here is CH12 and here is MEL. I think this is the region here. It's highlighted in black, in red. And you can see there's a big change. Peaks that are there are just not there anymore. And then you could also plot some of these differential sites. So what we're doing here is we're subsetting our log to then plot with pheatmap, and we're subsetting based on ones that significantly change or not. And so this is another way to start to drill down and see what the changes are in your data set and maybe what fold changes there are. So in this case, we have we're getting our R log values out with our assay. So that means we now just have a count matrix. And then we look at our differential analysis. So this is the DE-seq2 object that's undergone differential analysis. It's the results. We'll ask which ones have a p-adjusted value of less than 0.05, so they're significant, and they're not NA. Okay, so we're asking, is it NA? And then we negate that with the exclamation mark. So we're asking, is it NA? If it's true, then it will become false. And so we'll extract out anything that isn't NA. And then we're getting the log2 fold change that's absolute, that's greater than 3. So this is big fold changes. And we subset our data set on this. So at this point, diff contains a bunch of names of very, very significantly changed genes. And then we can use the in function or the in operator to subset our R-log matrix, which is our counts matrix, based on them having the same name as the significant genes. We can chuck it in pheatmap, and you can see that we have two main groups. We have ones that are upregulated and ones that are downregulated, right? Yeah, which makes sense because we're just looking for significant. And we're also looking for a log fold change of greater than 3. So these are going to be quite drastic changes between the two data sets. Okay, so that's kind of like the things that we did before. You can also do things like clustering, but I think the key points here is you can do much of the same kinds of analysis, pass these count data backwards and forwards, isolate gene sets and analyze them in similar ways. Okay, so the next thing is to focus on other ways to simplify your data down. And one of the key ways to do this, which we got covered a little bit at the end of the ChIP-seq and the ATACseq sessions, is Soggy. And so what Soggy does is it allows you to generate metaplots, which by this we mean just like averages over a given set of regions for the signal. So I think this one is cut site footprints around CTCF motifs. And this one might be TSSs. Okay, but it doesn't really matter, but we're going to show you how to do it. And so to do this, yeah, this is Tom's package. It's available on Bioconductor, but some of its latest features are not in the current release. At least I don't think so yet. So what we can do is actually just install it from GitHub. And so if you ever want to install anything from GitHub, people often host their packages. You can just run the code install_GitHub, and then you say where it's from. And so this is pretty much how you're installing the packages that contain our teaching in it. You can install it in the same way. And so to do this function, you just need to have DevTools. But once you have that, you can then load in this new version of Soggy. Okay, and so to be able to use Soggy, we just need some regions that we care about. And so what we're going to do is take our we're going to add some additional information to our G ranges that contain our DE-seq results. And so in this case, we're just going to add some categorical information. So we're creating a one vector initially, which is if it's upregulated in mel. Okay, and we're just going to be asking that question. Is it significant, and is the fold change lower than -3? And then we'll ask the question, is it down in mel? So we ask the same question, significant, and then it has to be greater than 3. And then from these two, we can use if else to create significance vectors. So we ask, is it present? Is the name present in this? Is the name present in this? And if so, you give it a label of up in mel, down in mel, or other. And then we can add this as a column to our G ranges. Okay, so why do we need is it not NA even when CTCF? Okay, so when you ask the question, is it less than 0.5? What do you think NA will return? Okay, I'll show you. Okay, so we have a vector and it has 5, 6, 7 NA. Okay. If we ask, say greater than 6. Okay, so we've asked a logical, and one of these is true, but this comes back as NA. And so when you try and index with an NA value, the indexing will get upset. And so we add in this additional step where we say and greater than 6, and we can do this. Okay, and now it works. Okay, and so that means if we then put this in an index operation, we'll get the 7 back. If we don't have that last one, then we get back 7 and NA again. So it's just cleaner that way, because otherwise you'll just have NAs following you around everywhere. And then at some point, the NA will be there and you won't know where it came from. And you'll have to go back through your data and carefully figure out where it is, because it's breaking whatever operation you're trying to do. Okay. So the other thing we're adding is breaking our groups into breaking signal strength into 2 groups, those that have high or low signal. Okay, so these things seem kind of arbitrary, right? What we're doing, but they kind of demonstrate how you can add this additional information that you might care about, something that you found interesting, or maybe you only want to care about the really big peaks or something like that. Or maybe they're peaks that overlap with something else. And so there's an easy way to go from your results, your DE-seq results, and then you add some extra columns with metadata in it. And so in this case, we're adding in some information about whether it's a high or a low peak. And so this is going to be based on the base mean. So if you remember, the base mean is the average signal across all samples. And we're generating quantiles based on this. So if it's like high in the quantile, it will be high, and if it's low, it's low. And then we're just adding an extra group based on this strength, whether it's high or it's low. Okay. And so we have this custom annotation now for strength. We also have this significance column as well, which is new. Cool. And so with this extra information, we'll get to that. But first, we'll just draw some metaplots with Soggy. And so with Soggy, we can use the region plot function. You'll need some kind of score information coming in, so we can use a bigwig. So here we're using the bigwig, and we're focusing on CH12. Okay, so we're saving that as CTCF bigwig. And then we're going to run region plot on CTCF bigwig. And we're giving it these ranges. Okay, so we're giving it our granges object, which has all this information. And we're saying style point and format bigwig. Okay, and distance around 750. So what this means is when you each peak has a different width, right? And so that is less useful if you want to, that it makes it hard to map equivalently if you're going to average across the genome. So when you say point, this means it's going to focus on the middle of the peak, and then it's going to do 750 base pairs up and downstream of that middle of the peak. And so if you have a really fat peak and you have a really thin peak, it's still going to center on that peak. Okay, and so when you do this and you do plot region, you can then see this really nice peaky CTF signal. Okay, and so this shouldn't be surprising. If you look at CTCF peaks, you see a lot of CTCF. It's a good control. If you don't see this, or you see some weird patterns around it, then that could also be informative. Soggy is built in Gplot, so you can customize it in exactly the same way as you would a Gplot. But what really makes Soggy quite nice is you can use these these summarized by arguments to essentially facet the Gplot object. So in this case, we're going to facet it by strength. And unsurprisingly, so this is our metadata column that we added, strength. And so you can see in the facet, this one is labeled high and this one is labeled low. Sorry, it's a little small, but you can go back to the Gplot course and figure out how to customize some of these sizes of things. We're just keeping it simple. But you can see the high peaks have more enrichment, low peaks have low enrichment, right? You don't have to do it this way. So this gives you the same result with this code. So we have already this list. We can create a list with our metadata information. As long as the names in the lists match up with the object, we can supply information externally by using the GTS argument. Okay, so this here is not using this column strength. It's actually using these vectors that are all the way back here. So these vectors have been turned into the column strength, but we can also, instead of integrating everything into the G ranges that we're using, we can just supply it from an external source here. Okay, so it does exactly the same thing. Oh, what's this one doing? Okay. Oh, yes, of course. Sorry. And so when we're doing this, by default, it looks at the row names for the matching. Okay. So each of these is a named vector, right? And to figure out which peaks correspond to these categories, it uses these names. And so it's going to assume that you're going to try and assign it or it's going to look at the row name. Okay. But we also have this additional column called name. And so you can specify which name you want to use. So if you have metadata in your ranges that you actually want to use instead, you could do that as long as there's matches. So this in combination with the GTS just controls how this matches your G ranges. And then the other thing that you can do is actually start to create multiple ranges that you care about to group the data. So you can use G ranges lists. And so in this example, we're going to look at TSSs and TTSs. So we can extract those out from the TXDB object, right? So we go to genes, extract out the genes from this, and we can look at this, the very first and the very end. And so what we're going to do is resize the genes object to be fixed on the start or fixed on the end. And so that will give us our TSS or the TTS. And we can supply both of these. Okay, and so all we're doing is providing this list into GTS. And we still have the original bigwig that we care about. And now we can look at the signal at TSS and TTS. We control coloring with the color by argument. So we can, you can summarize in one way, but you could color in a different way. It depends on how you want to do it. And you can also control faceting that way as well. So it's group by, summarize by, color by. Okay. And again, we can use still, it's all based in ggplot, so we can use any ggplot functions to modify what it looks like. So in this case, we've saved this previous object as P. We've got some new colors we want to use. And so we can then add the scale color manual and change the values of the colors and also update the theme. And so now we can have a more minimalistic plot with some different colors. And so the next thing we probably want to do is, instead of just comparing regions, we want to compare between our samples. So in this case, we can load in a second bigwig. And so we have the CTCF bigwig. And we can load it in. And this one is going to be loaded in as CTCF mel. And then we can combine these together. So if we have CTCF mel and CTCF CH12, and they're both a chip profile. Okay, and we save this as CTCF. And so then we can use plot region with CTCF, and they'll both be plotted together. Okay, and they'll just inherit the name of the bigwigs for this. And in this case, we're going to color by sample. So most of the names for all of this are very intuitive and make sense, I think. Instead of coloring by, you can also group by. So instead of it being on the same plot, you can facet it out in much the same way. Or you can combine some of these things. So in this case, we're summarizing by one of the metadata columns, which is SIG. We're also grouping by group and coloring by sample. And so this means the down in mel and the up in mel are at opposite ends, and the ones that aren't changing are in the middle. And you can really see the patterns at these peaks of what's happening. You can see a very low enrichment for mel for the ones that are down in mel, and the reverse pattern in the up in mel. So it seems that in this case, it isn't just, it's both there's a decrease and an increase at these peaks. So you can start to make judgments and inferments with subsetting things like this. Okay, so we used SOGI to produce plots of footprints at motifs in the ChIP-seq course, at the end of the ChIP-seq course. Okay, and so the idea of this was we could extract out all CTCF motifs and then look at the signal at these regions. There is, most of the time in data analysis, a reverse way to ask the same question to reinforce what you're learning, or or checking, or whatever research you are doing. And so doing the same analysis but in reverse is often a really good way of supporting your findings. And so in this time, what we can do is, if we look at CTCF peaks, is the motif occurrence really high at CTCF peaks? So this is the reverse question. And so to do this, we'll be using matched PWM. And so first, what we can do is use the seq logo function or seq logo package and function on the CTCF motif. So we're loading motif.db. This has the CTCF motifs saved along with all the other motifs. We can query it. So we're querying this database and looking for CTCF. This returns a list of potential motifs. We're going to extract out the first one because that's normally the top hit, the most accurate, up-to-date motif. We'll take that, and then we'll draw a seq logo. And so this is the traditional CTCF motif. We have this high region of Gs over here. We're starting off with a pair of Cs. And so we can use then match PWM to scan the genome for CTCF motifs. Then we can convert that to a G-ranges and then to an REL list using the coverage function. Okay. So we do match PWM. We look over this time we're just going to focus on chromosome 19 for this motif. We can then turn that into a G-ranges. Okay, so first we're going to do this into an I-range with this, and then specifically we're on chromosome 19. And then we do coverage to turn it into an RLE. Okay, this is, again, skill sets we've been using throughout the course. And what we get back is basically regions that are hits for the motif. And so we would expect the motif to be highly prevalent in CTCF peaks. So what we can do is then take this coverage here, this RLE, and we can plot it. And we're saying we're going to plot it specifically at CTCF regions where we've seen peaks. And we'd let it know that we've got an RLE list. And so we make the region plot, and then we plot the region. And when you do this, there's a massive enrichment of these motifs directly at the middle, pretty much, of CTCF peaks. compared to the surrounding regions. So this reinforces this idea that what we found is the motif is enriched at peaks, and the peaks are enriched at motifs. And so then you might want to be able to plot your CTCF signal in MEL versus motif occurrence next to each other. Okay, it's pretty straightforward to do. You have both data sets, but they are completely different data sets. Okay, so we have our motif plot here on the left, I believe. Yeah, and we have our chip signal here on the right. They're both showing at exactly the same regions, but they just have different signal. This is how often the motif is found, and this is how often you got a chip pull down. And so we're combining together these data sets by first subsetting our CTCF MEL dataset just to chromosome 19, so we have the same regions. We're then saving all the row names as null, so they're equivalent to each other. And then we just use the combine function. Okay, and so we have these two regions that have been through Soggy's plot. They're both Soggy region plots, and so then we can actually just plot them once they've been combined. Oh, we do have a question. Could you also do that if motifs are in Grange's object? Let me see. So it depends on your question. So could you also do this? So what we did in the ChipSeq, at the end of ChipSeq, I can show you. I know you weren't there for that. Bear with me for 2 ticks as I try and remember the URL. Cool. I believe it's the end of this section, but I could be wrong. It must be in here. Okay, so I can't find it right now because I can't remember exactly where it is. Maybe it's the beginning of this one. No, I can't find it. Maybe we removed it, and so that would be a problem. Anyway, so sorry about that detour. The answer is yes, but it would ask a different question. So what you could do is extract out all of the motifs, G ranges, but then you need and so once you have a G range, you need something to compare it to, right? And so that's something that you get a score from or a signal. And so in this case, we're specifically getting our score or our signal from the coverage of motif. So is the motif present or not, whether it's 0 or 1? And that's our score. And then we're using our chip peaks as the regions we care about. And so you could definitely do the same thing as here, where instead of looking at chip peaks with CTF signal, you could look at motifs with CTF signal. Okay, and so this is the kind of the two ways of asking it, right? We have chip peaks with motif signal, and then we have motifs with CTCF signal. And if you see a peak in both, that means really you have a motif that CTCF is binding too strongly, and you have a CTCF, which is CTF signal, which is found at motifs. It's kind of the same thing, but in opposite, but it shows that this kind of starts to point at like sufficiency and requirements. Okay. Cool. But yeah, so what we have here is instead of flipping them, we're actually only just changing the signal. So in both of these plots, we have the same ranges. These ranges are peaks, okay, CTCF peaks. And in the left, we have signal, which is how frequent is my signal, my motif there. And on the right, we have how, what's my chip signal. But because they're different signals, they're on completely different scales. So what you can do is use this parameter called free scale. And what it will do is it allows them to be on different scales and freely scale each facet. Okay, so in this case, we have a different scale here to here. But they have the same pattern, and that makes sense. And so what we've been doing so far is we've been using a metric. I explained the point metric. And so what this does is it allows you have a peak, and it will take the center of the peak and it will count outwards from the center of the peak. And so really the peaks are in the middle of this region. But we don't necessarily care about the nature of the peak, because if you have a really wide peak and a thin peak, they're treated as much the same. But for some features, that's an issue. An example would be a transcription, like a near a TSS and a gene, because genes have different lengths, but you might want to map a protein that binds to genes and map specifically how it binds across the gene. And so for that, you use a gene metaplot. But to be able to do that, you need to extract out all the information about the genes. Okay, so we'll grab all the genes. We get rid of their names. And so now we have this ranges for every single gene. And so now, instead of doing style equals point, we can do style equals % of region. So that means every single region is scaled. And we're specifically going to be using this bigwig here, which you can download. But there isn't a link to where it is. Okay, you can Google this specifically. It's an ENCODE data set. That's why it's ENCODE. But it's polymerase 2 serine 2. And so the different modifications and phosphorylated forms of polymerase have really different roles in the RNA polymerase action, right? And so you can start by using plots like this, you can start to see it in action. So we're using plot region again. But when we set this up, if you remember, we said the style is % of region. And so that means every single gene has been scaled to be this pseudo length. And then it also adds in some extra, which is also scaled to that pseudo length. And so you can see a big peak at the start and a big peak at the end. And so this is most used for anything related to gene and patterns over the gene. And so we could also include our CTCF data in this in much the same way and compare it to POL2S2. And so in the first one, we have our POL2S2, which is, no, this second one we have POL2S2, the first one we have CTCF, and I think it's well known that CTCF is quite often near promoters. And so we can see that in action. Okay, and so we're grouping these data sets again together just using the combine function. So it's really easy to just start playing around with the data, create these region plot objects, and then start mixing and matching them to see what they look like. There is also a function to start making these heat maps, which I mentioned at the beginning. We are going to overrun by like 5 minutes. Apologies, guys. But we're nearly there. We're just going to touch on some heat maps. And so first off, when you're plotting heat maps are more memory intensive than some of the other plots we do, because actually graphing them out can take a lot of memory. That's because there's lots and lots of little bins, lots and lots of little pixels, and the value in each of them have to be calculated and then actually plotted. So quite often what we'll do is we won't do it for like the whole genome or anything like that. We'll focus on some regions we care about. And so in this case, we're going to subset our CTCF objects to those that are only up in mel. Okay. So we're going to say their name is present in up in mel. And there is a function within Soggy called plot heat map that allows you to get to grips with this. Okay. And so this is a nice first pass. You already have your Soggy object, and you can start to get an idea of what it looks like. However, this is quite rudimentary, I'd say. The plotting heat map functionality was quite minimal when Tom made it. And so, yeah, this is just showing some examples between our different samples. But Doug, who is part of the BRC, former Alice Lab member, took what Tom had with Soggy, and then also got inspiration from what the folks at Deep Tools are doing, and then started to create a more advanced method for making these heat maps. And this is called Profile Plier. And so this is, again, another bioconductor tool, and it's really great. And if you ever need help, you can just ask Doug, and he's on campus, and you can email him super easily and he'll respond. So it's definitely a tool that you can use and get help with. And so the nice thing about Profile Plier and the way it's built is it's built with Soggy in mind, and it's also built with Deep Tools in mind. So if you're a Deep Tools aficionado, you can easily import Deep Tools objects into Profile Plier. Or if you're not, you've never heard of Deep Tools, it doesn't matter. You can take what we've learned with Soggy and we can import that. And the way we import the Soggy object is by doing as Profile Plier function. This will convert it into a range summarized experiment, which we've met before. And so it contains all this information within this object. You can subset it just like any other range summarized experiment. You can index it in the same way. And so in this case, if we just wanted to look at chromosome 1 in this region, we could create the G ranges and subset the Profile Plier like that. And we can also update the mcols associated to subset it as well. So in this case, we're subsetting based on whether the mcols is an up or down regulated. But really, what you want to do once you create this object is then start generating heat maps. And so there's a function called generate enriched heat map. You do this on your Profile Plier object, and what you'll get back is a nice set of stacked heat maps. And on top, you have the Metaplot, similar to what you would have got from Soggy. Okay. You can also perform clustering within this using the cluster ranges function. Okay, and so this shows you that there's one group that's pretty high in CH12 and another group which is mostly high, another group which is much smaller, which is high in mel, which I think we saw earlier. And so this does hierarchical clustering and gives you a nice summary. And you could then take that output and then put it into this, into your final heat map. And so in this case, we have the ordering. We're using order by function, and we're ordering it by the hierarchical order. And you can see that we have these two groups. We have this one group over here, which is our mel group, and then we have this one group over here. Okay. And there's ways to extract this information and then start to do extra things with it. This is it. I'm also just gonna, before we get off Profile Apply, if you click on the link on Doug Burrows' name, he has a wonderful little workflow for working through this. If you click on here, there's a roadmap data workflow. And this is a step-by-step which shows you some of the stuff you can do with it. Okay, and it's all loading in. But I'm going to scroll to the bottom to show you what's possible. You can do stuff like this. You have all these different data sets. You have clustering or groups. And so it separates out the metaplot. And then you can have additional annotations on the side, depending on what class that region is. Okay, and so you can get pretty wild with this. And it works pretty well and can make some very cool plots. Look, you can see it, more and more annotations keep being added. Yeah, so it's a really nice package for being able to show your chip data in an aggregated way, and you can present a lot of complicated ideas in a simple plot. Okay, so that is us. That is us done for today. Feel free to stick around if you have questions. We covered a lot in the session today. So I wouldn't be surprised if you do, but I also wouldn't be surprised if you just need to go and lie down for a bit. If you do need help and it's after hours, remember you can always go to our GitHub issues. So you can just click on getting help here. And you might even find that you have the same issue that some others have had. Okay, and you can write it in here, and we'll try and address these issues for you. Okay, and of course you can always email me, but we encourage it to end up here. Cool. Well, hopefully I'll see some of you next week for genome assembly and or single cell. I'll be sending an email out about that very shortly. Until then, have a great weekend.